4. Reciting prayer. 5. Photo session. 6. Plenary sessions. 7. Announcement. 8. Closing remarks followed by break, duhur prayer, and lunch. Then parallel session.
Moving right along, we will sing our national anthem, Indonesia Raya, and Hymn of University of Jember. Audiences are welcome to stand up. Please be seated.
The next agenda, we will have welcoming speech by Committee Chair's report, Associate Professor Dr. Dwi Wahyuni MKes. Please welcome.
Tes, tes. Assalamualaikum warahmatullahi wabarakatuh. Bismillahirrahmanirrahim. Distinguished guests and dear colleagues. First of all, on behalf of the organizing committee, I am delighted to welcome you to the Faculty of Teacher Training and Education, Jember University, Indonesia. Also, I would like to thank you very much for embracing the time to attend ICOPAMBS 20 and 21. Let me say how pleased I am to see such a great number of participants here. About 197 participants have registered. About this tremendous level of interest truly reflect the wide concern of natural science, mathematics, education, medical science, pharmaceutical, health education, and agriculture education. I'm also pleased to note that the majority of our participants come from various universities in different countries. India, Thailand, Philippines, Malaysia, but within and beyond Indonesia. I would like to address my special honor to our keynote speaker, Associated Professor Dr. Rames Nahimutu, Biomedical India, Associated Professor Dr. Mahana Priya, Associated Professor Dr. Katri Paikamta, Thailand, Associated Professor Dr. Mori Wikomani, Associated Professor Dr. Nakamani, and Professor Dr. David, Indonesia. Welcome to the Faculty of Teacher Training and Education, Jember University, Indonesia. Our faculty include 5 departments. Mathematics and Natural Science Education, Social Science Education, Liquid Science Education, and Education Science. My faculty, Mathematics and Natural Science Education, my department, have 5 program study for undergraduate study program, Mathematics Education, Science Education, and Physics Education, IPA Education, and Biology Education, and 3 postgraduate study program, Mathematics Education, IPE Education. As we all know, ICOPAMBS 20 and 20 is the 3rd international conference on natural science, mathematics, and education. The first conference have successfully 155 articles published in Scopus Index Journal. The second conference have successfully 111 article publishes in Scopus Index Journal, ICOP. I do hope the conference become our annual agenda, making possible continuous development of knowledge. What is more, I would like to express my sincere appreciation to one, Director of University of Jember, Bapak Iwan Taruna PhD. Two, Dean of the Faculty of Teacher Training and Education, University of Jember, Bapak Profesor Dr. Bambang Supeno, MBD. Three, the organizing committee. Four, the father of idea ICOPAMBS, Professor David, PhD, winner of Sinta Award, who have shown the profound support in making this conference possible. I believe that you will have an exciting meeting here in Jember University. Please use this opportunity to interact with it ID to the full in this unique opportunity and action you experience and ideas for the future. Finally, I wish all the success to the conference participant, and let's me once again extend my warm welcome and regard to all participants. I wish you an inspiring and enjoyable conference. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you very much, Associate Professor Dr. Biwahyuni MKes. The opening remark will be delivered by Dean of Faculty of Teacher Training and Education, Professor Dr. Bambang Supeno, MPD. Please welcome. Profesor Dr. Bambang Supeno.
Okay. Sorry, I do hearing. Assalamualaikum warahmatullahi wabarakatuh. Ladies and gentlemen, first of all, we have to thank God of His blessing and mercy so that we can come and join the third international conference on physics and mathematics and biological science on the so-called ICOPAMBS at the Faculty of Education, FKIP, Jember University in the years 20 and 21. For two class, that is on the 4th and 5th of September 20 and 21, 2021. I could have and condition. The third ICOPAMBS 2021 is still conducted online via Zoom, meeting due the pandemic COVID-19. Ladies and gentlemen, as the Dean of FKIP Jember University, I would like to thank the presenters for the time, chance, opportunity, ideas, knowledge, and skill that will be given and shared to the participants of ICOPAMBS 20 and 21. Besides, I would to also to thank all the members of our committee who have been working hard to facilitate and the international conference to run well. Apart from this, I word of thanks also directed to the participation on audience joining the international conference. I do hope that this annual conference will run well and will be critical and fruitful for all Utya, especially to participation. Thank you. Saya langsung membuka ya ini Dek siapa? Dek David.
Ya,
Oke.
betul.
Okay. In the name of God, Allah Subhanahu wa Ta'ala, today on Saturday, the 4th of September 2021, I officially open this international conference. Good luck.
Please give applause.
And Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much, Profesor Dr. Bambang.
You welcome.
Ladies and gentlemen, the next agenda is Uttering Prayer that will be led by Dr. Hobri MPD. Please welcome.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahirabbil alamin. Wassalatu wassalamu ala sayyidina Muhammadin asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Allahumma antas salam wa minkas salam wa ilaika ya'udus salam. Fahayyina rabbana bis salam. Ya salam, sallimna ajma'in waj'alna min ibadikas salihin. In the name of Allah, the most merciful, the most kind. Ya Allah, I beg thee of all good things which pertains to this day and days hereafter and seek. The protection from all evil which pertains to this day and days hereafter. Ya Allah, you are peace, and all peace is from you, and all peace return to you. O Master of power and glory, in conjunction of the Third International Conference on Physics and Mathematics for Biological Science, ICOPAMBS 2021. Guide us among those whom you guide. Grant us health and pardon among those whom you grant health and pardon. Allahumma inna nas'aluka salamatan fid din, wa afiatan fil jasad, wa ziyadatan fil ilmi, wa barakatan fil rizq, wa taubatan qablal maut, wa rahmatan indal maut, wa marfiratan ba'dal maut. Allahumma hawwin alaina fi sakaratil maut, wa najata minan nari wal afwa indal hisab. Ya Allah, unite us in this conference and provide us with a full knowledge, with a strength and the right effort to make the appropriate action. Ya Allah, Firilai, we ask you for the best supplication and the best success and the best deed and the best reward. Allahumma ya Qawiyyu ya Aziz ya Fa'alul lima yurid. Ya Allah, you are the most forgiving and one who loves forgiving. Therefore, forgive us and wish all participants in this conference and its related meeting a very pleasant and productive. Amin ya Rabbal Alamin. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina adzaban nar. Wa sallallahu ala sayyidina Muhammad wa ala alihi wa sahbihi ajma'in. Walhamdulillahi rabbil alamin. Summa assalamu alaikum warahmatullahi wabarakatuh.
Waalaikumsalam. Wabarakatuh.
Thank you, Dr. Hobrik MPD. Ladies and gentlemen, the next agenda is photo session. We would like you to turn on your webcam so we can see your lovely face. Please stand still and keep on smiling. The committee will take the screenshot.
Agenda selanjutnya adalah photo session. Dimohon untuk semua partisipan untuk membuka dan mengaktifkan webcamnya untuk bisa kami screenshot wajah-wajah yang ada di Zoom. Mohon untuk tetap bersabar dan memberikan senyum terbaiknya.
3, 2, 1, smile. The next slide. 3, 2, 1, smile. The end of slide. 3, 2, 1, keep smile.
All right, thank you. And the next agenda is watching profile video of University of Jember and profile video of ICOPAMBS 2021.
Universitas Jember berdiri pada 10 November 1964 di Kabupaten Jember, Provinsi Jawa Timur. Dengan mengemban cita-cita seperti yang tersemat dalam sesanti, Karyori Naras Abuko Budi, Gapuro Mangesti Aruming Bawono.
Cita-cita tersebut dituangkan dalam visi misi Universitas Jember, yaitu unggul dalam pengembangan sains, teknologi, dan seni berwawasan lingkungan bisnis dan pertanian industrial. Universitas Jember saat ini telah memiliki 15 fakultas, 103 program studi, 1.260 dosen, dan 33.928 mahasiswa. Selama 56 tahun perjalanan mengembangkan Tridharma Perguruan Tinggi, kini Universitas Jember menjelma menjadi perguruan tinggi terakreditasi A yang berada di peringkat 22 nasional dan telah memperoleh piagam penghargaan sebagai penyusun laporan keuangan terbaik ke-1 kategori Satker PNBP DIPA di atas 200 miliar. Berdasarkan analisis SWOT, Universitas Jember menetapkan tujuan dan sasaran strategis yang ditetapkan ke dalam tahapan-tahapan yang terukur di segala lini sumber daya yang dimiliki untuk meraih universitas yang unggul dan ternama di era revolusi industri 4.0. Mahasiswa Universitas Jember telah mengukir prestasi baik di tingkat nasional maupun internasional dan terus berupaya untuk selalu meningkat dari tahun ke tahun. Dalam sektor pendanaan, baik penelitian, pengabdian maupun publikasi jurnal ilmiah, terus mengalami peningkatan tren sehingga mampu mendorong tumbuhnya realisasi Tridharma Perguruan Tinggi. Di sisi lain, Universitas Jember juga meningkatkan kerjasama dengan mitra baik dalam dan luar negeri. Alumni Universitas Jember telah banyak tersebar dan berkiprah di dunia kerja, baik di dalam maupun luar negeri. Hal ini tentu tidak terlepaskan dari kualifikasi dosen yang terus berupaya ditingkatkan agar dapat beradaptasi dengan tuntunan dan perkembangan zaman. Universitas Jember memiliki 4 kampus, yaitu Kampus 1 yang berada di Jember, Kampus 2 di Bondowoso, Kampus 3 di Lumajang, dan Kampus 4 di Pasuruan. Semua kampus telah memiliki gedung dan fasilitas pendukung yang memadai dengan total 1.935 unit dalam upaya meningkatkan potensi pendapatan. Universitas Jember merencanakan 9 divisi usaha yang akan dikembangkan yang terdiri dari divisi usaha berbasis bioteknologi, unit pelaksana teknis, kompetensi bidang ekonomi dan bisnis, kompetensi sumber daya manusia, layanan kesehatan, kompetensi bidang agribisnis dan agroindustri, kompetensi kepariwisataan, keteknikan, serta pertelevisian dan perfilman. Berdasarkan pendapatan dan kebutuhan yang telah dipetakan, Universitas Jember memproyeksikan pendapatan dan belanja dalam 5 tahun ke depan yang terus ditingkatkan dari tahun ke tahunnya. Keberadaan Universitas Jember yang mampu berbicara di tingkat internasional disertai dengan otonomi yang lebih besar, diharapkan mampu menjadi faktor pendorong kemajuan Jawa Timur bagian timur di bidang pengembangan SDM pada khususnya dan Indonesia pada umumnya. Dengan kondisi yang demikian, maka Universitas Jember siap menjadi BLU, kampus merdeka dari Universitas Jember untuk Indonesia Jaya.
UNED siap BLU.
Good morning, ladies and gentlemen. Especially good morning, Forum FKIP. Welcome for joint meeting together in this International 102nd ICOPAMBS 2020.
The Honorable Rector of the University of Jember, Vice Rector for Staff and Finance Affairs as representative today, the Honorable all Deputy of Deans of the Faculty of Teacher Training and Education, the University of Jember, who is joining in the session in the same room today. The Honorable Student Speakers, yang telah hadir hadirat dan sekalian, second Dr. Amos Cibutu from India, the third Associate Professor Joanna V. Sarana from Philippines, the fourth Professor Ismail from Turkey. Loving and gracious Allah Subhanahu wa Ta'ala, you are indeed the giver of all good gifts, and we thank you today for all your blessings.
Today, I will talk about optimization models and the methods in machine learning. So, that is outline of my talk. This is first I will give a brief introduction, then I will consider 4 different types of problems.
Good morning, everyone. Sorry, I was on mute. So greetings from the Philippines. How are you, everyone? I hope everyone is safe despite the current situation that we are in. So let me just share my screen. Okay, I hope everyone can now see my screen. For this morning, I will be talking about technology in education.
Today, so I am going to discuss about the therapeutical categorization of bacteriophages for against carbapenem and pollution resistant enterobacteriaceae. So this is my field of interest. I used to do research in the field of antimicrobial resistance and bacteriophage therapy. So nowadays, we lost all available antibiotics in the world because of overuse, misuse, and improper use of antibiotics.
As mentioned by moderator,
I'm working a lot on combinatorics and graph theory, but later also I'm working on combinatorial education. Now, our conference, apart from talking and discussion about the basic sciences, now also we would like to share the application of basic sciences in education.
Ladies and gentlemen, welcome to the plenary speaker session.
In this first plenary speaker sessions, we will have Dr. Ramesh Nachimutu and Professor Dr. Andes Davik, MSc, PhD. This session will be led by Mrs. Siti Murtia, SPD, MPD, as the moderator. We have 2 hours for the first plenary sessions. Moderator, time is yours.
Okay, good morning, everyone. Thank you for finding time and joining us today in this conference. Very warm welcome to all of you, all the participants and our honorable speakers. I feel privileged to extend my warm welcome to all of you who are present in today's occasion. My name is Siti Murtia, and I am the appointed moderator for today's talk. Before we get started, just a little bit housekeeping. I will read to you some guidelines for today's presentation. Guidelines number 1, that we will have 1 hour for each speech, which will be divided into 2 segments, which are presentation, and the remaining time we will go for the Q&A section. The guideline number 2, if you have any questions during the presentation, please kindly type it down in the chat room. I'll bring them up during the presentation if necessary. The guideline number 3, if you wish to deliver your question during the Q&A time, please use the raise hand features and you can deliver it directly to our speakers. And the last guideline will be to keep the quality of sound functional during the meeting. Please kindly mute yourself when you are not shared, but remember to unmute it when you wish to share. Okay, so now without further ado, let me introduce you to our first keynote speaker. Ladies and gentlemen, joining us today is a renowned researcher with over than a decade experience in his field. His eminent work is in medical microbiology, especially antibiotic resistance and bacteriophage therapy. And he also creates a antimicrobial resistance awareness program. And initiates bacteriophage research and therapy in India, especially. And he had completed one DST SERP project, two consultancy project, and having 11 international collaboration with Turkey, Norwegia, Australia, Malaysia, United Kingdom, USA, South Korea, China, and Russia. That's among of them. Currently, he spends most time in Department of Biomedical Science, School of Bioscience and Technology, Vellore Institute of Technology, Tamil Nadu in India. So, ladies and gentlemen, please give us a round of applause for Dr. Ramesh Nasimutu. Good morning, sir. So please, we will have 1 hour for you, sir, to give your presentation, maybe 45 minutes, and we will use the 15 minutes left for Q&A session.
Okay.
The time is yours, Dr. Ramesh.
Thank you. So is my audio clear?
Yes, sir.
Okay, is my slide is moving one to other? On just a minute. Let me check once again. Is it moving on other?
Moving, sir.
Moving. Okay, so my voice is clear. Is my voice is clear?
Yes,
Yes.
clear, sir.
Yeah. So, Salam Alaikum, Salamat Baki. Thank you so much for inviting me for your third international conference. I'm very happy to present our research findings to this conference. This is my third talk. I'm very proud to be in this ICOPAMBS. So today I'm going to talk about the exploring small live animal, animal infection models to study the efficacy of phage therapy. So as I mentioned, I am working in the field of antimicrobial resistance and bacteriophage therapy for the past 2 decades. Means that we are focused on antibiotic resistance because of we lost all available antibiotics in the world. So we need alternate therapy. So that alternate therapy is the bacteriophage therapy. Even though we have some few options, but that won't be that much efficacy. Therefore, we decided to go on the bacteriophage therapy. So this bacteriophage therapy, when you start to work with this bacteriophages, isolation, characterization, and another thing we need to go with the in vitro analysis and in vivo analysis. So in vitro, it's everyone may have a good kind of facilities can do this work, but when we talk about the in vivo, that is tedious job and need a lot of funding and facility, a lot of technical things. Like that, we need to have a large animals, and the amount we spend is too quite higher. So therefore, what we decided, why don't we go for a small, the live animal infection model? So that is what we decided, and we started to working on it. And very soon it is going to publish. So I'm here giving you and presenting you the unpublished data. So we'll enter into this the title. So the bacteriophage therapy uses live bacteriophages for the treatment of bacterial infections. I hope everyone knows that the bacterial infections very well. Like for example, I can give you the Salmonella typhi, typhoid fever. So the typhoid fever is caused by the Salmonella typhi bacteria. So to kill the bacteria, we need some antibiotics. Okay, and also we can say urinary tract infections. The urinary tract infection is caused by gram-negative bacteria, E. coli, Klebsiella, and like. So to tackle those, we need antibiotics. So for all the bacterial infection, we need antibiotics. Because what happens, we misused, overused, improper use of antibiotics. We lost all available antibiotics in the world. So therefore, as I told, so we enter into the bacteriophage therapy. So the phage therapy, as I mentioned, so live. We are going to use the live bacteriophages. So bacteriophages is nothing but it is a viruses that kills the the bacteria. The bacteria is a food for the bacteriophages. It depends on the bacteria. Okay, and it records, it use all the machinery of bacteria. Once what happens, it go and attach to the surface of the bacteria, the cell wall of the bacteria, and then inject the genetic material inside the bacteria, and then it use the enzymes, machineries, everything, and then it develop itself, inside the bacteria. And finally what happens after the development of the bacteriophages inside the bacteria, it bursts out the bacterial cells and comes out again. It goes and find some other bacteria, and for example, there is a single bacteriophage become a 80 or 90 bacteriophages. So that 80 to 90 bacteriophages again go on to the 80 different bacteria and kill it. So it is a genus specific bacteriophages. Okay, bacteriophage is the genus specific. The bacteriophages will go on stick only on the specific genus and species and strain specific. Okay, for example, if you are isolating a bacteriophages, it belongs to E. coli, Escherichia coli. It kill only the Escherichia coli, especially the species and strains. Okay, it will not kill the other genus of Klebsiella and Pseudomonas or Streptococcus. Okay, but if you prescribing antibiotics, for example, it's a narrow spectrum antibiotics of gram positive or gram negative, it kills all gram positive or all gram negative. Or if you are prescribing broad spectrum antibiotics, it kills all the normal flora which is present in our body. So our human body in it estimates, for example, if you are weighing 60 kilogram of your body weight, the bacterium starts microorganism. I can say microorganism starts 2 kilogram of weight. Means that much microorganism is present in our body. Okay, that does lot of the support. It gives a good protection from the pathogenic microorganisms. Okay, so if you are misusing antibiotics, what happens? It kills the all normal flora which is present in our body. So therefore, what we need to, so when we go with the bacteriophage therapy, for example, the patient is infected with E. coli, it go and kill only the E. coli. Okay, it will not kill any other, even the other species, other strains of E. coli. Okay, means it is the advantage for us. Okay, so that is what now the 110-20 years before we are using it, but as soon as we find out the penicillin antibiotic, we stopped using the the bacteriophages. And after 1928, the Alexander Fleming, he discovered this antibiotics. After that, we keep on finding out new antibiotics, and past to 30 years, we did not find out any new antimicrobial agents. Okay, means that we lost everything. Okay, so it is not possible to get the new antibiotics. So we are going with the bacteriophages therapy. Okay, so it is not new to this. It's 120 years back we are using it. Only thing is we still the Georgia. Okay, the former Soviet Union regions called the Georgia. So Georgia, they are using the bacteriophage therapy, and around the globe there are many companies are there for bacteriophages. In India, we have a company called Gangajan Biotech. Okay, so there are many scientists working in this field to find out the new bacteriophages. Okay, to treat about the bacterial infections. So here the bacteriophage therapy, as I mentioned, it's a live bacteriophage we are using to treatment of bacterial infections. So from the clinical perspective, if you see it, the study result of the in vitro models vary significantly. Okay, when you are getting a result from the in vitro, that is very different to the clinical side. So therefore, the preclinical studies are very important to characterize the bacteriophages. So the using large animals in preclinical studies such as rat or mice needs ethical standards and costly, as I mentioned. So to maintaining the large animals, it's too costly. Okay, also we need to spend more money. Okay, for example, if a study with a single bacteria and a single phages to the animal studies, it costs nearly Indian rupees to 2 to 3 lakhs. Okay, so if you are going with a small animal models, it's very cheaper. That is what. And also, if you are going with a large animal models, we need a lot of ethical approval. Okay, we need to, the committee should may approve and may not approve, and we need large spaces, large facilities like that. So that is with this basic, the objective. Why don't we why don't we go with the small animal models, which is approved in the scientific community? So now we have to find out the simple animal infection models to the efficacy of bacteriophages. So before go to that, I like to give you show you some examples. So the death. Okay, just a minute. I'm having just second. I'm sorry to just a minute. There is some I have. Sorry for that. Yeah. So when I ask, what is the deadliest disease in the world? So you can say that, this and cancer and AIDS, you can say, but this is the prediction of this, WHO in the 2050. Okay, so there will be a, you can see the antimicrobial resistant infection is that higher. No, there are 10 million people. Their estimation, to infect and kill 10 million people. After that is a cancer, and then is a diabetic, and then is a diarrheal disease, and then is a road traffic accidents, measles, cholera, and tetanus. So I can say it's worse than the corona. So antimicrobial resistance and the superbugs. It's called as when the bacterium is resistant to all available antibiotics in the world. That is called superbug. So that bacterium is worse than the corona. Okay. Now the estimation, the future estimation with the antimicrobial alone, you can see this bar. So now why? That is what we are using this bacteriophage therapy. So how to choose simple whole animal infections models? Okay, so universally it should be accepted universally, especially for the infections model, and easily maintained in the laboratory conditions. Okay, and easy to handle and cost effective, no ethical approval record, and easy availability. And it should be available. No? It's not, for example, if it's available in a particular country, I need to go and get it, and it is not an easy job. So it should be available very near to you, and suitable for inducing battle infections, and a convenient experimental model, and mimic the large animal models, and supports phage multiplication. So these are the some of the the criteria we should follow while go for a small infection model. So the common animal models used in phage therapy. So we were using the rabbit and mice and hamster and G. malonella and rat and G. pepper. So these are the animals we were we are using it, and we were using it. Okay. So we can see here these are the small animals. Okay. The small animal, nothing but now here I'm going to discuss about two small animals. One is the Galleria malonella and Canadropix elegans. So these two are the small animals we are going to discuss. So the Galleria malonella, the greater wax moth, is a moth of the family Pyralidae. The J. malonella eggs are laid in the spring, and they have 4 life stages. So the wax swarm, of the greater wax moth have been shown to be excellent model organisms. Okay, there are many studies, and okay, there are many scientists around the globe. They are using this Galleria malonella as the infection model. So they are studying. They are using this worm to study the sterility pathogen, bacterial pathogens, or any kind of pathogens. Okay. So another side, so C. elegans, you can say as C. elegans, Canadropix elegans. It's a free-living transparent nematode about 1 mm in length. You can see here 1 mm in length that lives in temperature soil environment. C. elegans can be easily infected by bacteria, fungi, and virus, inducing lethality to non-lethal infections. Okay, that is the advantage here. So I hope still my audio is clear. Okay, yeah, I'm continuing. So the G. malonella, you can see here. This is the G. malonella. It's a it's adult worm, the cream in color. So this we have to inject in the last left pro leg. Okay, so you can see here this is the last left pro leg, because the reason behind of injection of this using this last left pro leg, because here if you are going in between, it may damage the intestine. Okay, that is why in the last left we have to inject our bacterial pathogen or bacteriophage or any, for example, if you want to introduce any compound, you can do here. So the artificial diet for the maintenance of Galleria malonella. So the Galleria malonella, the diet. This is the diet. The composition, media composition. So we need 120 gram of honey, 120 gram of glycerol, 200 gram of milk, 60 gram of yeast extract, 100 gram of wheat germ, 100 gram of wheat flour, and 120 gram of wheat bran. So these are the composition of the diet for the G. malonella. This is very easy, and it's a easy availability. Anywhere you can buy this, all these things. Okay. So we are actually, we have a collaborator, Dr. Prakash KV from the Bangalore, Karnataka. They used to send us the worms whenever we require, and also we can culture from our lab alone. Okay, you can see here the worm in the artificial diet. Okay, these are the worms. So the advantages of G. malonella. So as a model host, G. malonella larvae have several advantages. Okay, so they possess a semi-complex cellular and humoral innate immunity. That is very important, because it should mimic the human body. Okay, that is what we are using here, and shares remarkable similarities to that mammals, and low maintenance cost. As I mentioned, the media and all, it's very cheap and very easily we can get it, and low maintenance cost, and ability to obtain large quantities. Okay, if you, for example, if you want to use large animal models, 10 or 20 or 50, it's not an easy task. But here, the worms can be n number we can produce, n number studies we can go. Okay, because it gives large quantities of these worms. And then fast reproduction time. Okay, so it's very fast. For example, if you are waiting for mice and rat and the monkey and this thing, so it's not an easily the reproduction is the very it takes huge time. But here it's a very fast reproductive time and ease of inoculation. Okay, when we deal with the animals, that is very difficult. Okay, but here and injection and giving our test compounds or battle or anything, it's very easy and used without major ethical concerns. Here, there is no ethical approvals required to use this now to go for a infection models. Okay, so this larvae can be kept at range of temperature from 20 degrees to 40 degrees Celsius. This is the advantage. Here, there is not much required of specific instruments like 20 degree, 25 degree like that. So minimum 20, maximum 40 degrees, 22 degrees Celsius. So it's allowing experimental procedures to mimic mammalian physiological conditions. So this is what it's record. So what are the ongoing research in our lab, especially the with the Galleria melanella? So the phage bacterial interaction study, you can see here. I think I can use the find the option. Yeah. So the phage bacterial interaction study, and then in vivo biofilm clearance by phage treatment, and then the safety and efficiency trials of E. coli, Klebsiella, Pseudomonas, and Staphylococcus phage. So we are using these 4 bacterial pathogens to test the G. melanella. Okay, and then bacterial virulence analysis, whether what is the most the virulence factor of the particular pathogenic bacterium, and therapeutic efficacy of phages. That is very important here. So these are the 5 different research we are doing, especially in G. melanella. So when you talk about, this is the paper we published in a BMC microbiology. So the therapeutic potential of bacteriophages targeting gram-negative bacteria using G. melanella infection model. So this is the first study I can say in the world of using bacteriophages to the G. melanella. But bacteria, they were used already. But bacteria and bacteriophages, this is the first time. So we designed some, this study design. This is our study design. You can see here. So G. melanella model, and we have 4 different sets: set 1 and set 2, set 3, and set 4. Set 1 consists of control and saline, because we are mixing the phages with the saline. That is what, whether the saline has some effect on the worm or not. And then set 2. Set 2 is the control and bacteria alone. Here, because we are using bacteria, okay. And set 3 is the control and phage, okay. So phage alone. And then test 4 further divided into 2. One is single dose and multiple dose of bacteriophages. Here, nothing but we are going to infect the worm with the bacteria, and we are introducing our bacteriophages, whether the bacteriophages protecting the worm or not. That is the objective of this study. Okay. So here the single dose infected with bacteria, the concentration is 10 to the power of 8 CFP per ml. Okay, treated with single dose of bacteriophages. Okay, and here multiple dose infected with bacteria 10 to the power of 8 colony-forming unit per ml, and treated with multiple dose of bacteriophages. Then the concentration is 10 to the power of 4 plaque forming unit per ml at 0 hour, 6 hour, 12 hour, and 24 hour. So this is the difference between the single dose and then multiple dose. Okay, I hope you got it clearly. And I'll show you the worms. Yeah, the study result. You can see here. This is the creamy white. It's a live and healthy worms. You can see here. This is the healthy worms. The creamy white, and you can see the dead or infected one. This is the infected, dark brown, black. This is the infected with the bacteria. Okay, pathogenic bacteria. Okay, so the bacteria, you may ask me. So the bacteria we are getting it from the diagnostic laboratory that we have already MOU with them. So they used to give us the clinical pathogen. For that, we need to clear ethical approach. Okay, we have more than 2,000 plus clinical pathogenic bacterium in our laboratory. We have a BSL2 facility, BSL2 facility to use this kind of work. So you can. This is the difference between the healthy and the infected one. Okay. So as I pointed out earlier, the injection, the the injection of the pathogenic bacteria. Okay, first in the last left prologue. This is the last left prologue. So if you inject here, it will damage the intestine. Okay, that is what the advisable region is the last left prologue. Okay. So the result, when you come for the result, so the activity of E. coli phage, okay, E. coli phage. So here, don't confuse it. I'm here using Escherichia coli phage, means that the phage, this Escherichia phage will kill the Escherichia coli, okay. So we are going to infect the worm with the Escherichia coli, and then we are going to give the bacteriophages after that. Let me see the result, how it's. So this is the control, okay. So the pink is the control, and there is no death happen, and then the blue is the bacteria. So after 48 hours with the bacteria alone, there is no treatment here. Here, the worm is died because the pathogenic bacteria infected the worm, okay, and then it killed. So next, the green is the bacteriophage. So there is we just injected only the bacteriophage to the worm. The bacteriophage didn't kill the worm, means that there is no effect to the bacteriophages did not have any side effect to the worm, means that it's a kind of clear. Okay, it's not required to worry about to use this bacteriophage as a therapeutic purpose. This is the aim objective here. And treated, this second one is the treated one. See, bacteria are now, when we injected the worm with the bacteria, the worm killed after 48 hours. And when the worms treated with the bacteriophages, the worm is killed and the reactivity is reduced after 96 hours. Okay, but in the control, the phage alone, then nothing happened because the worm is clear. Okay, clear. So means that, so the 75% here we got 75% survivability and all dead within 96 hours. Okay, means that it indicates that for this bacteriophages, single dose of bacteriophages is not required. It requires multi-dose of bacteriophages. This is our objective here. So activity of Escherichia phage, the same bacteriophages, here we are using multiple doses. Okay, multiple doses. So here you can see this is the control, and treated, green is a treated dose 1. Okay, and treated dose 2 is red, and then here treated dose 3 and treated dose 4. So the bacteria alone, it killed 48 hours, and single doses. Okay, you can see 96. It killed after 96 hours it killed, and multiple. See here the yellow one. Okay, it's a for dose 4. So completely killed, means that the worm is recovered. Okay, the multiple doses. There, single doses cannot, the worm cannot be recovered because it required multiple doses. When we given the multiple doses, here the recoverability of the worm is 100%. So after that, what happened? We crushed the worm and then checked. Okay, the phage only, there won't be nothing. Bacteria will not be there. The bacteria alone, that's you can see the 10 to the power of 8 9 CFU, and here dose 1 still bacterium is there. Dose 2 still bacterium is there, and dose 3 and dose 4 there is no bacterium. Means that the bacteriophages kill the bacteria and recover the worm. This is the result for the Escherichia phage. And next one is the Klebsiella phage. So Klebsiella phage, the same study, and you can see here the bacterium, it infected the worm and it killed 48 hours. And the control, the phage does not have any effect to the worm. And then when we go for a test, okay, treated with the Klebsiella phages, here we got 100% survivability achieved at single doses up to 96 hours. But lesser movement was noted, means that it recovered it, but the movement of the worm is lesser because it needed a multi-dose of infect. So here the multi-doses, when you go for a multi-doses, you can see the 100% recoverability, okay, of the worm. Okay, so this is the yellow. Okay, the 4 doses 4, and doses 3 also, doses 3 and for red and okay, yellow, you can see here, and the pink is the control. So 100% recoverability. And when you go for enumeration of the bacteria, so there is no bacteria found in the dose 3 and dose 4, means that the worm recovered because of the bacteriophages killed the bacteria. So ours in the same the phage number 2, Klebsiella phage also, it required multiple doses. So another bacteriophage is the Enterobacter phage, single dose, and same here. And here we got a 80%, okay, 80%, and rest is the dead because because the concentration of the bacteriophage is needed more. Okay, that is what. So the effective with the survival of 80%, rest all dead within 96 hours. Single dose is not sufficient to achieve 100% survival of larvae. Okay, that is, and again we go for a multiple doses. So here when we go for a multiple doses, okay, and the recoverability. See in this page, bacteria pages, doses up to there is a bacterium is present in the doses 3, and there is no bacterium in the doses 4. Means that here this, so in the study what we concluded, the each and every phases has their own therapeutic potential. So we need to find out. Okay, that is what. So the doses 4 it completely killed, and the 100% survivability of this worm. Okay. So next, so what we decided, why don't we go for multiple and cocktail? Okay, why don't we mix? For example, if a patient got infected with more than 1 or 2 bacteria, so why don't we go for a cocktail? Cocktail is nothing but we are going to mix the 3 different bacteriophages in a single shot, and then we and at the same time we mix the 3 bacterial pathogen and injected to the worm, and then we introduce the bacteriophages, cocktail bacteriophages with the same concentration, and then we injected. So when you see here, when you go for a cocktail, the until the doses 4, the bacterium is present, and when you go for a doses 5, okay, completely killed. Means that we can go for this bacteriophage therapy when the patient got infected with multiple infections. Okay, this is our concept. Okay, so the survival rate is, there was only 60% survival rate, and there was improvement in the larval survival after 4th dose and 5th doses. So this is our conclusion in this study. So, and then experiment number 2. Sorry, there is some. Yeah. So the next study, I hope I want to confirm my audio is clear, right? Shall I continue?
Yes, sir.
Yeah, thank you. Yeah, that is all. Yeah, the second experiment, I think I have a time. Yeah, still I have half an hour. The experimental setup, study 2. So what we did was, this is another, so far we were trying with the gram-negative bacterial infections, and another infection caused by the gram-positive bacterium, that is the Staphylococcus aureus. Methicillin, there is a 2 type. One is the methicillin sensitive Staphylococcus aureus and methicillin resistant Staphylococcus aureus. So here what we decide, why don't we try with this Staphylococcus aureus for the bacteriophage therapy, and also the biofilm former. That is our main objective in this study. So the biofilm forming Staphylococcus aureus, the antibiotic cannot penetrate to the biofilm, and it cannot kill the bacteria. Okay. So here we need a big question: how to save the patients, those who infected with Staphylococcus aureus. Okay, especially the biofilm former. So why see, we decided, okay, why should we should use the, why not we go with these bacteriophages to the Staphylococcus aureus therapy. So when we decide and now design, so we made in this study, what we did was, we made we want to infect the worm with the biofilm forming Staphylococcus aureus. Okay. So what we did was, we used the tooth bristles, okay, the sterile tooth bristles with the bacteria. Now after 21 in a microwave plate to make a biofilm, and over the 24 hours, and after the biofilm forming, the bristles we used to inject. That is what, because we to the injection we can inject the suspense of the bacteria, but in this study we need a biofilm forming bacteria to infect it. That is what our concept. That is what we made the tooth bristles. That bristles overnight grown with the bacteria, especially the Staphylococcus aureus. Okay, and overnight it form a biofilm, and then we washed with the buffer, and we have, you can see here, this is the tooth bristles we are injecting here, but it's very small. Okay, it's very small. It is not very easily handled. We need some practice. So after the biofilm formation on the bristles, that is injected in the last flow leg. Okay, after that we introduce the bacteriophages every 2 hours, 5 hours, 8 hours, and the recovery of the worm was observed for next to 48 hours. Okay, here what happened? The biofilm formers were infected with the worm, and then we introduced the bacteriophages. We check whether our bacteriophages kill the bacteria or not inside the worm. That is our main objective. Okay. So So and also for the control, we also given the antibiotics. Okay, so after saline 100 mg per kg and vancomycin 20 mg per kg body weight. In the here we chosen the average weight is 300 to 500 mg of wax form. Okay, this is our concept, and also the antibiotic treatment and the sterile tooth. For control we have given sterile toothbrush. Okay, and then the antibiotic have given as a control, and we just administered after the antibiotic we introduced the bacteriophage 2. So the result when you see, you can see here, this is the biofilm infected worm. Okay, the blue one is the biofilm infected worm. It killed after 72 hours. Okay, and antibiotic treated worm. This is the red one is the antibiotic treated worm. It also killed in the after 72 hours. The worm is killed. Okay, because the antibodies cannot treat because the bacteria is formed in a, so it is in a biofilm form. It formed a biofilm. Therefore, the antibiotic cannot pass through it and kill the bacteria, and it killed the worms. Means that biofilm killed the worm, and also the even antibiotics also it did not kill it. And when we go with the bacteriophages, 3 doses, here we use doses 3, so the recovery of the worm is 96 hours. And then control, okay, you can see the purple is the control, green is the bacteriophages, red is the antibiotic treated worm, and blue is the biofilm infected worm. You can see here clearly the bacteriophages treated is recovered, and there is I'll show you once again. Yeah. So here what we suggest was the multiple doses of bacteriophage. If you are prescribing the multiple doses of bacteriophages, definitely the 100% recovery would have done. But here we have used only doses 3. So, but our main conclusion whether the bacteria is recovered because of the antibiotics or not. But here it's not recovered because the bacteria is already formed in a biofilm. So the antibiotics cannot work and perform in the biofilm forming and the bacteria. So next, so C. elegans, okay, Canodropid elegans. So this is another small live animal used. So what is the ongoing research in our laboratory? The safety and efficacy trials of E. coli, Klebsiella, Pseudomonas, and Staphylococcus phages. Okay, whether these are the safety and efficacy. Okay, where the isolated the E. coli, these bacteriophages, safety and efficacy with this study or not. And then bacterial virulence analysis we did, and then therapeutic efficacy of phages and phage interactions by fluorescence imaging. This is another new studies we are going to publish very soon. So the cultivation and maintenance of the C. elegans. So the C. elegans is a Bristol N2, so wild type, is maintained in the laboratory on nematode growth medium. So this C. elegans can we get from the C. elegans Genetic Center, CGC, okay, in the US. Those who wish to do and research in this C. elegans, you can be a member, and for education purpose they are giving free, and for academic for education we need to pay $100, and for research purpose there I think it's more than $100. And for example, if you want to use this C. elegans, for example, if you want to use in Indonesia, okay, you need to find out who are working in this C. elegans, and anybody has this the license. Okay, we cannot simply work and publish this C. elegans study without the permission from the CGC Center from the US. Okay, so we can it also the easily availability from here. I got from our collaborators from Tamil Nadu alone we have. So if we are getting the nematode, the C. elegans we are using for our study. So the isolate called as C. elegans Bristol N2, so wild type. Okay, so the bottle of media can be stored at room temperature for several months. It is very easy maintenance. And the E. coli OP50. Actually, the food for the C. elegans is the E. coli OP50. Okay, E. coli OP50 is the food for this C. elegans. So it is the E. coli and grow well. Okay, so this is the first what we need to do. First we make a media. From that media we have to grow the E. coli OP50. To that we need to, after forming the the culture, growing the bacteria, we have to introduce this C. elegans. Okay, I will show you the video of how it to look like. Okay, so the seeded plates stored in an airtight container will remain usable for 2 to 3 weeks. We can keep until 2 to 3 weeks. And the worms tend to spend most of the time with the bacteria because it requires food. So the a limited bacterial lawn is desirable because it allows for easier observations. Okay, it can be, the C. elegans is transparent and can be visualized using a dissecting stereo microscope. Okay, or inverted microscope. If you have a basic microscope, you can easily visible this C. elegans. Okay, the moment, and I'll tell you what is the which stage we should use for a infection study. So you can see here, this is the nematode growth medium, okay, the regions of the nematode growth medium and M9 buffer. So this nematode growth medium is very easy and cheap, and it can be obtained any scientific communities. And then the M9 buffer for studying purpose. For example, if you want to study the pathogenicity, you can go with the microliter plate and the M9 buffer, and then introduce the worm inside, and then you can go with any kind of studies. And here I'm just focusing the bacteria, but you can go with any kind of studies. Okay. So this is the adult worm, L4 stage, and this is the eggs you can see here. So the general stock maintenance steps, nothing but the C. elegans stocks can be maintained between 60 degrees Celsius and 25 degrees Celsius. So most typically at 20 degrees. If you have a cooling incubator, that is very good enough. And the C. elegans grows 2.1 times faster at 25 degrees Celsius than at 16 degrees Celsius, and 1.3 times faster at 20 degrees Celsius than 16 degrees. So this variation in growth periods can be useful when planning for your experiment. Okay. So worm storage and the recovery. So the worms can be stored in a liquid nitrogen in the stage of L1 and L2 stages. So I'll show you the stages. So for example, if you are working with this worm and you want to continue this work after a month something, okay, you can easily store in a minus liquid nitrogen, minus 196 degrees Celsius. Okay, and you must be careful that the stage should be L1 and L2. Yeah, you can see the movement of the C. elegans. So this is the L4 stage. This is the L4 stage. You can see here in the microscope. You can see here. This is the X, and this is the L1, L2 stage. Once again, I'll repeat it. Okay. Yeah. You can see here the stage used here. This is a L4 stage, and this is L1. This is egg 0.1 mm in size, and L1 and L2 and L3. And the study for our study purpose, we should use the L4 stage. So when you are hatching in your laboratory, you will be very easily get to know that what is the stages of this form. Okay, it is very easy. It's easy to maintain. Okay, that is what. So another one is the experimental setup. So nothing but previously we were using the Galleria. Here we are talking about only the C. elegans. Okay. So age synchronized L4 worms were exposed to the test pathogens in a 96 well plate containing M9 buffer in each of the wells for phage efficacy assay. So this is the 96 well microliter plate we used, and the M9 buffer we already added, and then the worms we got it from Pereira University from our own collaborator. So the L4 stage worm is picked 15 worms, okay, approximately 15 worms, and a liquid-based assay was performed for evaluation of effect of phage on bacterial clearance. Okay? This is our concept. So there we used Galleria. Here we are using C. elegans. That's it. And the remaining phages and the bacteria is the same here. So the Staph aureus culture 10 to the power of 8 CFP colony forming unit per ml was added to the well containing nematodes and allowed to infect for 8 hours. Okay, we added the worm in the M9 buffer, and then we just introduced the Staphylococcus aureus, the pathogenic bacterium in a C. elegans, and then we left for until 8 hours. Okay, so on phage 10 to the power of 9 PFU was introduced to the well, and the nematode score was recorded for the interpretation. So as soon as you introduce the bacteria, it go and infect the C. elegans, okay, worms. So when it infects, and after that we are introducing the bacteriophages. So the bacteriophages go and infect the, and here we mentioned the mono and cocktail, nothing but single bacteriophages and multiple bacteriophages. This is the difference between mono and cocktail. So when we see the result, okay, you can see the result here. The phage 1 and 2, the blue and then yellow, it reduced to after 7 days. Okay, and here the SA2 and the SA5, you can see here. So this is dropped already because it directly killed the worms, and there is no prophylactic over there and killed it. And then this is the cocktail. So when you see the cocktail, 100% survived. Even after 7 days, the worm is the animal, the C. elegans is recovered because of the multi doses. So from our study, we are ensuring that the single, and when compared with the single doses, the multiple doses of bacteriophage is efficient for the bacteriophage therapy. In the all the level we have tested in the even with the Galeria, even with the C. elegans, okay, even with the inotrosis. So it's everything is supporting the more than 5 doses. Okay, that is what you can see here. Even if you are using bacteriophages, it's reducing for single doses. But when you are increasing to the multiple doses, the recovery of the salience is higher. Okay, so as you can see here, the worm was stored for 7 days and TD50 was calculated. The mean reduction of bacterial load for phage treated phase 1 and phase 2 showed 70 and 65% efficacy, and the cocktail of phages showed 100% efficacy with no death observed for 7 days. You can see the result, 100% showed for a phage cocktail. And phase 1 to mono, it gives only 70 to 65% because it requires additional the phage therapy phage load. So the bacteria and the phage effect on nematode life cycle, especially we need to talk here. The phage cocktail showed more efficacy. No death of the worm was observed for 7 days. Single dose of phage also had better effect on the bacterial clearance. So the for cocktail, 7 7 Staphylococcus phages used. Okay, this cocktail method showed no effect on nematode cycle. Okay, so this the bacterial treated well showed the absence of life cycle, so that infection by Staphylococcus aureus has adversely affected the nematode growth. Okay, so the bacterial treated well showed death in 8 hours after 8 hours, because when introduce the bacteria to the worm, there is no prophylactic, there is no antibiotics there, so it will automatically it kills. So another very important studies. This is our at present we done the study and we are planning for the publications. So the fluorescent labeling, okay, fluorescent labeling of phage and bacteria. So I think this is a new study. They did with the bacteria, but not phages and combined infections to the C. elegans. And you can see the image here. So this is the worm, the same worm what you have seen in the video. The same worm was infected with bacteria and another phages. Okay. So here the fluorescent labeling, I hope you might have seen the fluorescent microscopy with the bacteria something. Here the worm with stain, the DAPI is a stain. The stain, the bacteria, what we did was we stained the bacteria in the dye, and then we infected with the worm, and then we did the microscopy, fluorescent microscopy. You can see here the bacteria. Okay, the bacteria entered to the worm, and it is now you can see here clearly. There's all the bacteria. Means that the worm is infected already with the bacteria. It will be killed. Here in the second image shows worm with the stained phages alone. Here the bacteria alone and the phages alone. Whether we want to know whether the bacteriophages entering to the C. elegans body or not. That is very animals. Okay, whether it's infecting or not. Okay, whether it's going inside or it's remain outside. Or, that is our objective. And what we find out was, okay, when we dyed, when we stained with the bacteria, and here is a bacteria and it's stained with the phages. So you may ask me what is the difference? Why we are using 2 different colors? So when we infect this bacteria and then bacteriophages, inside we doesn't know what, we cannot differentiate it. Here the dye is called cytothertin. Okay, cytothertin, green fluorescent dye. Here is a blue, DAPI blue. Okay, so DAPI. So, and you can see here next image, worm with stained bacteria. Bacteria is a DAPI blue color, and then the phage, the green color. So now you can then you can see it, the blue color. This is the bacteria, okay, and then green is the bacteriophages. So we also confirm that the bacteria and the bacteriophages entered inside the worm, and we can it can confirm that. So both are entering inside. Okay, and the worm with only the stained with phages. Okay, you can see here the body, inside the body you can see very small the staining, and excuse me. So worm alone stained with cytothertin, so without phage. You can see here we stained the worm Okay, we stain the worm with cytotectin. It's a green fluorescent tag, green fluorescent dye. Okay, green fluorescent dye. There is no bacteria, there is no bacteriophages introduced, and it's clearly easily visible. This is the L4 stage of the worm. Okay, and here also the same worm with stained with the phase 1. You can see here inside is the pages are there. This is the stained one. Okay, stained one. It is already there. So means that it is confirmed that it's confirmed the bacteria and the pages is entering the worm, and it can be studied easily, and we can produce the result what we are expecting it. Okay, so these are the advantages of using the small animals, especially in the bacteriophage study. Even you can go use with any pathogenic studies. Okay, that can be done, and only thing you must have a experience of using this fluorescent microscopy. Okay, so very easy. Fluorescent microscopy is very easy. Only thing you must know how to dye this stain. And in India, we started a society called the Society for Bacteriophage Research and Therapy. And because we need to create a awareness to the researchers around the globe. And you can see here, she is the Dr. Elizabeth Cutter. She was from USA, and she was the chief guest of our conference. And the reason behind of doing this is well motivated to the students. Okay, all the students can you can at the age is 83 now. He is running 83. At the age she on the way to US to India, she came and attended our conference, and very active. Still she is doing research, and we have good collaboration with her. And those who wish to become join as a member to our society, we have a site. This is the website. You can go through the site and you can be the member. And also we started a journal, Society, okay, International Journal of Bacteriophage Research. Those who wish to work and want to submit the paper, you are welcome. Okay. And this is the not only we are doing research, and the result on the paper is not useful. Maybe may not useful for the society. Okay, our the utmost the useful is whichever the result we got, it should reach the society in the language, the local languages, and the people must know the reason behind of antibiotic resistance is the people doesn't know the difference between antibiotics and then other medicines. Okay, simply without going to clinicians, directly going to the over the counter and getting the antibiotics and taking it. But that is not advisable. Okay, the antibiotics should be taken only with the prescription of the clinicians. Okay, so that is what we created a organization in India, the Indian Initiative for Management of Antibiotic Resistance, and we created in the year of 2017. I am the national joint coordinator and the campaign coordinator. I have conducted this this awareness campaign around India. Okay, we covered all the part of the India, and we made different languages, and we made the script and transferred to all part of the people. You can see here we are made all the peoples and disseminating the information, and we what to do, what to not do, like that we have. So this is a thing we did. Even we gone for a primary school there. The kids must know this. Okay, what is happening with what is the difference between antibiotic and other and drugs. So this is my research lab. So as I already when I visit, you are very well welcome. We have a good fund, but I can host you in my laboratory. So we have a team of big team, research team. And thank you so much. Thank you for your great, kind and great listening. I hope this enlightened everyone. And this is a new field, and everyone might have, at least to get some few points related to the bacteriophage therapy and the AMR. So if you have any questions, please ask me. I'm ready to answer any kind of questions you want to ask.
Thank you, sir. Ladies and gentlemen, please give a round of applause for Dr. Ramesh. Okay, thank you, sir, for giving us a valuable insight of the topic. I can see already the valuable contribution for your research, actually, sir, because we have to admit that antibacterial resistance is main challenge, if I may say like that. It's the main challenge in health industry, along with the emergence of novel viruses like the one that we have today. So with that, ladies and gentlemen, we will go ahead to take some time to questions. This meeting is now open for sharing. Once again, please use raise hands feature if you wish to share questions with us. or you can also type your question in the chat room.
Oktopa and Jepti Prihatin.
Right.
Yes.
Okay, okay. We have one question for Buyekti. The time is your Buyekti.
Thank you, Bu Dia. Good morning, Mr. Ramesh.
Morning.
I'm Yakti Prihatin from Biology Program, University of Jember. Will I think your research is very amazing. By using small invertebrate, you can prove that fakes can reduce or overcome bacterial infection effectively. And you use many techniques with colorful staining. It's very amazing. I yes, and but now I'm not asking you about fakes, but I want to ask you about artificial diet for Galeria melonella pyralidae. I'm interested with the artificial diet.
Artificial diet, yeah.
Artificial diet, yes.
Yeah. Yes,
The food of the insect.
Food, yeah.
Yes. As we know that the insect can decompose plastic effectively. 100 larvae can eat 92 milligrams polyethylene. So, yeah, yes. And my question is, why you use wet as a major component, and is artificial diet durable, and how long? Thank you.
Thank you. It's a good and excellent question. Actually, the worms can eat plastics. Already it's reported. Everyone knows this. Even we have tried to. We have given the worms, especially the single-use plastics. So the single-use plastic, when we given within 1 hour or 2 hours, the complete group of the worms eaten up. Okay. So it is very interesting. And again, what we did was, why don't we get the intestinal bacteria to go for plastic degradation? So we also in the year of 2008, I have we a team of members, my PhD colleague, we did a study in plastic degradations. So we were used to different kind of bacteria, and it was 10 to 20%. But when I see this, this information, this worms, Caloria marinella, when it eats the plastics, and we know the idea was enlightened. Why don't we isolate the bacteria that is especially present in the gut? Okay, we just crushed the gut, and then we started isolated few bacteria, and what happened after the isolation of bacteria? Unfortunately, another pandemic is started, and we could not continue the work. And related to this diet, especially if you asked, the diet is very easily available and very the short period. Now we can get 5 to 7 days. We'll get it, and after that, even we can maintain it more than 20 days, 20 to 25 days. But what happened? We have to pick the small worms, and then again we introduce the new media, and then it takes another couple of weeks. Okay, and then we based on the study, we have to go for hatching of this worm. That is very important, and the only thing is very easy and cheap maintenance. Okay, rather than using the large models, large money, here it's very cheap. Okay, so the maintenance cost, when you see, it's less than 1,000, okay, 1,000 Indian rupees. So it is very easy. Yeah. I hope I mean, yeah.
Is there any feedback, Buyekti? Maybe you want to say something to add your question?
Yes, thank you.
Okay, already answered
you.
and Brandy. Okay, thank you, Bu Yekti. As for your information, Dr. Ramesh, Bu Yekti focus on the on research on Ulat Sutra. How to say it?
Wild sick and sick
Silkworm.
moth.
Silkworm.
Silkworm.
Yes.
Silkworm.
She focuses on developing artificial diet in silkworm. So,
Yes.
thank you, Bu Yekti. Maybe another question for from participants. You may raise hand or write in the chat room.
Octava S.
Oh.
Pamungkas.
Okay, please. Mr. Octava, if you want to say your question. Yeah. Oh, okay. While waiting for other participants to ask question, if I may ask question, Dr. Ramesh. Well,
Yeah, please. You're welcome.
From biology, but my interest is on mycology actually. So
Mycology.
yes, I'm not aware with the virus and so on. But one thing that I want to ask you, and hopefully it's not out of context. Hopefully it's not out of
No problem.
Yes. Uh, uh, with this prolonged pandemic, sir, in the span of 2 years, we already had 5 or 6 variants of coronavirus. So, what is exactly present in its genome, sir, that make this virus so easily to mutate?
Since the mutation is usually does with all the microbes exist in the world, because we experienced the whole population of 7 billion people of the world, they are experiencing now, and there is no awareness to this field, especially biology. Only the people working in biology, they know what is mutations, what is happening with the bacteria, virus, protozoan, and fungi, and etc. So now when the people knows, everyone, all the 7 billion people are talking about mutation. Yeah, it's normally does with all the microorganisms. Only thing now the corona got it spreaded, and then it reaches other part of the country, and the geographical location, the temperature, and then the temperature plays a major role, and then the host, where it lives, and these are the possibilities of this mutations. So this is quite common in biology field. Okay, so it may be different or maybe threatening for other peoples, but this is very quite common. But and also the social media now. Initially, the 2 or 3 decades before, we don't know the information happened in other part of the country. But now, every second, we can get information from all part of the world. Okay, this is what happening. This is what's spreading it. So this is the things happening. So only thing, be have a vaccinated and be safe and take a healthy food. Nothing won't happen. We'll be recovering very soon.
Okay, thank you, sir. So one question again. I will take from participants, if any. Okay. If I may, well, one of my friend, my colleagues, apprentice, he is unable to join us. Oh, we have one from
Miftahul Firdaus.
Miftahul Firdaus. So I'd like to ask, viruses in the public perception are organisms that have a bad impact. When used in medicine, how do you convince the public or patients that fake therapy is safe?
Excellent questions. Really, I appreciate the question. this is the big challenge for the scientific community that that the biologist knows that the normal flora, right? So bacteriophages can be considered as a normal flora. That is not a pathogenic virus. That is not a virulent. And our human genome already contains 7% of the viral DNA. Okay, so as soon as we did the human genome sequencing project, out of that we have seen that 7% it shares already with the virus genetic material. And also it is not a pathogen, it is not a virulent, and it is like a normal flora, and also it is present in our gut already. Okay, in our human gut, our human body already contained the bacteriophages already. So we were using 120 years back, okay, as during the World War I and II, and even the Soviet Union, still there is a bacteriophage therapy center is there in Eliava Institute. It is in Georgia. So there are many patients are saved using these bacteriophages, and there is no harm at all. It is also when compared with antibiotics, this is best treatment options. It does not contain any side effects. Okay, only thing at present, since we don't, nobody's not worked after stopped with the 120 years back. Now the scientific community around the globe, we are generating the data. We are finding out. We are getting a lot of proof that we are getting the concept and proving and through the scientific papers and publications. Okay, so even many of the countries now started with bacteriophage. They are allowing the even very recently last year this pandemic, we had a review that, so why don't we prescribe these bacteriophages for the secondary infection for especially the COVID patients. Okay, so after a few months back, the Food and Drug Administration has announced that the bacteriophage therapy can be given to the patient infected with COVID-19, especially the secondary infection, secondary bacterial infection. Okay, so now everyone expecting the proof of scientific data. Okay, that is what around the globe, all the scientists we are working and generating the data that it should support for the therapeutic purpose of the bacteriophage. So obviously the question is excellent, and it's the biggest challenge for us. Okay, it's a bacteriophage is the normal flora. It cannot be mentioned and said by the virulent and pathogen. It is a safe to use. It is a live good option to go with that.
Well, Miftahul Firdaus, ini Bapak atau Ibu ya? Do you have any feedback? Miftahul? Well, apparently, sir, there's still a lot of work to be done to introduce this kind of therapy to society, I think. Okay, so maybe we can get another question from participant. Okay, so maybe one more question from me, Dr. Ramesh, if you may.
Please, whenever you can ask. I'm happy to answer.
Interesting, actually, very interesting. So some bacteria have defense mechanism to protect themselves from virus infection, such as whisper. Then, is it still possible to use fake as biocontrol in future? If so, how, sir?
Actually, the bacteriophages even can be used to. We are also working with aquaculture. We are focusing on aquaculture pathogen, and we are also planning for agriculture pathogen, and especially for bacteria. No? Since the bacteriophage can be used for only the bacterial infections. So we are not only limiting ourselves with clinical pathogens, we also focus on biocontrol for other aquaculture pathogen and agriculture pathogen too. So it can be used, and as I mentioned, it will not infect or kill any other strains, of the same genus. Okay, so these are very genus and species and strain specific. It will not kill any. It will not disturb the normal flora which is present in our human body. Okay, so it can be used for any kind of fields, even food industry we can use. Now in the US they are using the Clostridium, especially Clostridium bacteriophages. The food industry they are using the bacteriophages. and it can be used in agriculture application. As I mentioned, it can be used in even the veterinary background we can use, and aquaculture we can use. So anywhere, all the bacterial pathogen, we can use this bacterial therapy.
Very interesting, sir. So which one is more effective, sir? Lysogenic phage or lytic phage?
Sorry, not clear.
Which one is more effective? Lysogenic phage or lytic phage?
Lytic. So lytic always no. So the concept of the bacteriophage therapy is the lytic cycle. Okay. So lytic, if you go with the lysogenic, it will integrate and it will not burst out the cells. But if you go with the lytic cycle, it will develop and multiply and burst out the bacteria. That is our concept. We should use the lytic. Always, lytic phages should be used.
Okay, thank you, sir, for your insight on this topic. I will check once again from the participant. So no. Okay, so looks like that we covered all of the question. So, Dr. Ramesh, is there anything else you want to add before a wrap up, sir?
Yeah, here I like to since the biology students, I'm requesting all the students that don't try to get the antibiotics over the counter, and it should be whichever. If you have any symptoms, if you have anything, please go to the clinicians. Okay, directly do not go to the pharmacist. Okay, the pharmacist doesn't know about what kind of infection you have. Okay, even based on the symptoms, we cannot find out exactly what kind of bacteria, or whether it's a gram positive or gram negative. He doesn't know that. Okay, so please go to the clinician first, and then the clinician must do the diagnostics of the particular clinical samples, and then he or she has to find out what are the infection the patient got infected, and then the clinician should prescribe antibiotics. And here it's not only stopping here. Another important thing, you should use the full course of antibiotics what the clinicians prescribe to you. That is more important. Okay. So for example, if the clinicians prescribed antibiotics for more than for example 10 days, okay, so morning and night is a 20 tablet he used to prescribe. When you are buying it, please buy it 100%. Don't reduce. Just give me only 50% of the tablet. Okay. So please get 100% of the tablet what he prescribed, and then when coming to the home, please continue the complete course of antibiotics. Any medicine, it should be continued. Okay, it's not be stopped in between. Okay, so what happens because of overuse? For example, overuse of antibiotics. Okay, overuse in the sense the cattle farm. Okay, so we are using a lot of tons and tons of antibiotics to the cattle farms for development of the chicks and the pig farm, and then the cattle, all the the cows and buffaloes, these things, and then the poultry farm also. We are for prophylactic, we are giving tons and tons of antibiotics. What happens? All the antibiotics are reaching to the soil. That the soil contains microorganisms. When the microorganisms expose themselves in a high concentration of antibiotics, the bacteria become resistant. Okay, we may get the infections, but while we are using the agricultural product, we may inject the food contaminated with the soil. That soil may contain the antibiotic resistant bacteria or antimicrobial resistant microorganisms that may infect us. Okay, so it should not be stopped. And misuse of antibiotics. Misuse of antibiotics nothing but the antibiotics. Okay, for example, it is prescribed to the father of the family. He might have infected with gram negative, and the same after 1 week, the wife or other children may be infected, and they may why don't we go for a clinician? Let me use the same antibiotics what they prescribe for me. This is a misuse of antibiotics. Okay. So, and improper use of antibiotics. Rather than taking gram positive, if you take for gram negative, so if you are changing the spectrum, definitely it may increase the antibiotic resistance. So, be very careful that while taking with antibiotics, that is very important. And another important thing, I'm going to inform you that we lost all our available antibiotics in the world, even the last resort antibiotics. Okay, if you are if there is any clinician, you'll know that. So be careful. Thank you.
Thank you, sir. So no playing around with antibiotic. We have
Sorry?
we have to take it
Be
wisely.
careful.
We have to take it wisely. Okay, so now if I may conclude our first talk as one challenge that compromise humankind, antibacterial resistance needs immediate and urgent efforts to solve. One of the sources to overcome this problem is using bacteriophage therapy. With its advantage and disadvantage, one breakthrough that can be applied to test the efficacy of the phage therapy is using larvae to replace the use of expensive in vivo tests. So once again, thank you, Dr. Ramesh, for inspiring presentation. On behalf of all the conference committee, we wish you and your family always in a good health, sir. Thank you very much.
Thank you
Give a round
so
of
much.
applause, ladies and gentlemen.
Thank you so much. I thank Dr. Dafik and his all complete your conference team for given the an opportunity to give the talk. I'm very happy to be part of the University of Jemper always. I thank once again Dr. Dafik and his team, and also I thank my friend Dr. Ivan. I hope he might. Thank you all. Thank you so much.
Thank you, sir.
Thank you all. Thank you all the conference teams. Thank you.
MC langsung. Okay, well, ladies and gentlemen, and all honorable participants, now we've come to the second session of our plenary speaking. Already joining us, our respectable researcher, as well as teacher and mentor, who relentlessly give us example of professionality and passion in pursuing academic achievement and acknowledgement. He is among the 50 best national researcher awarded by Ristek Brin in the year of 2020 with Scopus document 194 with citation number 540 and Sinta National Rank number 17 and Sinta Affiliation Rank number 1. At the moment, he is the head of SIGAN Research Group and lecturer on combinatorics in mathematics education study program in Jember University. Ladies and gentlemen, please give a round of applause for Professor Dr. Andes Davik, MSc, PhD.
Thank you.
Before you
start it?
Before you start, Prof, I think I have miscommunication with the committee. You have 1 hour to present the paper and then 30 minutes to Q&A session, Prof.
Thank you. Thank you. Could I start, Ibu Siti Murdia?
Yes, Prof.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah wa syukurillah wa la hawla wa la quwata illa billah amma ba'du. Honorable Dean of the FKIP University of Jember, Chairwoman of the ICOPAMBS 2021 conference, Ibu Associate Professor Dr. Dwi Wahyuni, and moderator, all the participants today. I am very happy to be a resource person in this conference. And I would like to share about education now. As I mentioned in the list of my CV, I'm working both on combinatorics science and as well as combinatorial education. Could I share my slide? The slide here, it is written in the first slide. The title of my talk will be the student meta literacy improvement by integrating research-based learning and STEM education in developing a secure crypto system by means of a rainbow anti-magic coloring of graph. This working paper joint work with Zainur Rashid Ridro Ibu Arika Indah Kristiana and Ika Hesti Agustin have been working on the research-based learning topic since 2018, as well as working on the crypto system and rainbow anti-magic coloring of graph. But now, in my talk, I will bring a little bit in the form of the educational side, so that participant could also join a discussion later on about this one. And this topic also taken by some my PhD student to be their PhD dissertation. This is my outline of talk. We will talk about what is meta literacy and also what is research-based learning. Further, we will talk about STEM education. And finally, we will implement rainbow anti-magic coloring into a secure crypto system. And while we implement this concept in the classroom, and then we will analyze their meta literacy of the student. So this is the educational side of this topic, Lishan. Motivation, I believe all people already know that we are in the stage of society 5.0. Along with this, we have also fourth industrial revolution. So in this thing, we have the most digital era. Then we have a top 5 disruptive technology in the Industrial Revolution 4.0. The first one is Internet of Things. I believe somebody already used this internet for every side of life, even for having a food, for having a taxi, for studying, especially in the pandemic or COVID-19 outbreak, all the time use internet. Along with this Internet of Things technology, we also know about artificial intelligence. Artificial intelligence gain a lot of benefit in this real life. For example, we have a smart car, we had a smart mobile phone, we had smart city and smart computer and everything because of meaning the artificial intelligence. And also now the era we have conversational user interface, the using of robot, the using of chatbot, also the using of neural linguistic process, and also using other intelligent algorithm and intelligent programming software. This is the indicator of that we have in society 5.0, including digital marketing, data information security. All people aware about this kind of benefit, then we use all the time in this era. So digital literacy in the 21st century era or society 5.0 gave a lot of side of aspect, information, data, and content, teaching and learning, and self-development, also communication, collaboration. We all the time use a mobile phone, use gadget, and everything. As well as creation, innovation, and research is also taken over all the time by using internet software or internet platform and everything. So that's why we need to have a technical proficiency, as well as we also know about the use of Internet of Things will give us a bit risk about how secure the Internet of Things be used in the real life. For example, if we have a transaction, online transaction, we are asked to input the data from our a credit card, debit card, our detail of our address and everything, our number, even mobile phone number, phone office number and everything. So they need encryption and decryption process. It is part of the crypto system. So digital literacy in the 21st century also force us to know the digital safety or security. So these three of things involved in the data literacy, and this one is involved in the information literacy. And finally, in this one is also important that we should have it, that is the literacy of humanity. So literacy of humanity is we need to avoid, for example, data abusement, data hijacking, and everything. So we are talking now about meta literacy. So meta literacy cover all this, and meta literacy actually raised from the digital or information literacy. So what is meta literacy? So if student will think about this kind of topics, so that actually students should find out first the state of the art, the references. The meta literacy actually firstly introduced by T.E. Jacobson and McKay. They wrote a book entitled Reframing Information Literacy as Meta Literacy. So they published in the Communication in Information Literacy Journal. Again, Jacobson and McKay continue to write a book about proposing a meta literacy model to redefine information literacy and so on. So we have Kay and Stewart, we have Brogard, and we have McKay again. So most of them, the paper and books written by the two guys, Jacobson and McKay. So Jacobson and McKay gave a definition or concept development about meta literacy. So meta literacy is a literacy which promote critical thinking and collaboration in digital age. So the keyword here is a digital age. It means meta literacy, of course, different with the other literacy, but the meta literacy must of indicator based on the information literacy, which provide a comprehensive framework to effectively participate in social media and online communities. So meta literacy challenge traditional skill-based approach to information literacy by recognizing related literacy type and incorporating the emerging technologies. So again, the keyword is the technologies used. So actually, meta literacy aim to achieve the 21st century skill. What is the 21st century skill? We have a critical thinking skill, we have a creative thinking skill, we have a communication thinking skill, and also collaboration. Collaboration is important now the era. Without collaborating with other people, such as in this conference, if we do not have friends from other country, we couldn't invite some other invited speaker easily, for example, apart from Indonesian invited speaker, for example. It is very important. And also joint paper, joint research could be built without any any problem, because with the using of cloud meeting platform, for example, we can use it. So that's why collaboration can be done across country, can be done across province, can be done worldwide. So that's why people in the 21st century, they should have these four of skills. So namely 4 C's: creative thinking skill, critical thinking skill, communication, and collaboration. So finally, yes. technical problem first. Just a minute first. Yeah, okay. Again, I repeat again, moderator. Okay, let me continue. So, what is the main indicator of meta literacy? The main indicator of meta literacy, according to Jacobson and McKee, first one is produce, yeah, and also incorporate, and third one is use, the fourth one is share, and the fifth one is collaborate. So by means of mobile technology, either mobile phone or laptop or everything, and by means of the platform, it can be where open educational resources, or it can be open courseware, or also another ones. Just today era is very powerful and very popular. We call MOOCs, Massive Open Online Courseware. So actually, meta literacy has 5 main indicators. First one, produce, incorporate, use, share, and collaborate. For produce level, it has 3 sub-indicators. The first one, identifying the properties or characteristic of problem. Second one, obtain a breakthrough. The third one is develop or determine the stage, phase, syntax, or algorithm. And corporate, in general, it need people can be able to identify the pattern of solution, also can be able to generalize, can be able to use Internet of Things such as a software, a platform application to integrate the result. And also the third level of the main indicators, it is used that we need to have also sub-indicators. In here we have 4 sub-indicators. The first one is testing, analyzing, interpreting, and implementing. And the fourth main indicator is share. So as I mentioned before, the main thing in the meta literacy, student or learner can share whatever they have to enrich their product, to enrich their idea. Of course, by using Internet of Things, social media, OER, open educational resource, and also MOOCs, massive online open courseware, teaching platform, and others. And the fifth is collaboration. This collaboration is part of the 4C in the 21st century scale. So in meta literacy also, it has collaboration indicator. The collaboration are split it into a sub-indicators. Here, the first one is work with some other people by using Internet of Things, and also enrich the product or by requesting some suggestion from other, encouraging people to do more, to contribute the findings, obtain a joint work and product to publish, and finally determine a joint future work for wider society. So this is the main indicators of meta literacy. So if the researcher will talk about this one, will study about this one, of course, they should start from the main indicators. And then they can split into a sub-indicator. And finally, maybe they should develop an instrument. They should develop an teaching material or teaching and learning material or whatever. So they can run the study or the research nicely. For competency domain of meta-literate learner, also we have metacognitive. So actually, when people reaching a meta-literacy as a what achievement or ability, and it's implies that people having a metacognitive nicely. So what learner think about their own thinking, this metacognitive, a reflective understanding of how and why they learn, what they do and do not know, their perception and how to continue to learn. Also, the other domain is the cognitive. So meta-literate people, meta-literate learner, is apart from having the main indicator, they should have also four domain here. The first domain is metacognitive, and second one is cognitive, and the third one is behavioral, affection, and the fourth one is affection as well. So this one is important in the meta-literacy. So meta-literacy do not deal only with the information literacy. So if people ask me about what is the difference between meta-literacy and information literacy, apart from having the main indicator in meta-literacy, produce, incorporate, use, share, and collaboration, and meta-literacy also having the domain competency, yeah, they should pass the four domain of competency: metacognitive, cognitive, behavioral, and affection. So if people having a meta-literacy, they also having a humanity of literacy. So they just do not use internet of things. They also maintain the character. What is the character of having using internet of things all the time? For example, avoiding the hijacking, avoiding the internet abusement, data abusement, avoiding about carding and everything, yeah, and also inputting the illegal data, for example, also avoiding to have unauthorized data input, for example. So this is in general meta literacy. So meta literacy cover basic literacy, which is rise from other literacy, for example, science literacy, mathematics literacy, information literacy, technological literacy, and finance illiteracy. So the meta literacy, of course, beyond of those literacy. So if people having science literacy, mathematics literacy, information literacy, and then they have awareness about humanity literacy, and they are also having a four domain of competency, then these people already reach a meta literacy. This is a meta literacy. So in meta literacy, of course, we motivate people or student to be a customer, not only customer, but they should be forced, they should be motivated to be a producer. So not only download it all the time, yeah, you should have this word activity, uploading data, uploading a product or uploading something in the internet, and you can share to other people, you can share to your student, you can share to other colleges, and I believe your meta literacy already now done. So meta literacy, student and teacher interaction should use internet technology all the time. For example, using encyclopedia, using ResearchGate, for example, ResearchGate, Orchid, Mendeley, and using some researcher profile to browse their product, their paper, their publication by using Google Scholar, Cinta, Scopus, Pablon, and everything. And also we have now cloud storage by using SlideShare, LinkedIn, Dropbox, Google Drive, Overlip Editor. There are a lot of things that you should understand, yeah, about information literacy to have a meta literacy, yeah, in our real life. Cloud classroom also, we need to have this one. We need to have knowledge about using Skype, Cisco Webex, Clicks meeting, Blue Jean meetings. All those things are information literacy. And also, if we add up with the four domain of competency, then we satisfy the meta literacy indicator. So this one is meta literacy. Yeah. Okay. Yeah, I give it back to a moderator first. Probably there is a discussion. I don't want to continue and finish, and after that there is a discussion. Maybe we can talk about this one first. Yeah, because this is in the first session of my talk. Yeah.
Yes, Prof. We have one question actually from participant in the chat room. I will try to convey it to you, Prof. So
Yeah.
the question is from Ahmad Jainuri, Mr. Ahmad Jainuri. Um, in general, Mr. Ahmad Jainuri stated that there are public comments or public opinions that consider meta literacy is not working in solving the real problem in Indonesia, Prof. So what do you think about that, Prof?
Yeah, actually, we have two section, yeah, two sides about talking about skill or talking about meta literacy. Yeah. So when we talk about meta literacy, there are science literacy, mathematics literacy, and also social literacy, finance literacy, and everything. Ministry of Education already put the new a rule about the assessment, yeah, national assessment for the school. In this case, of course, they do not require about meta literacy of the student in the national assessment. What they require about the national assessment is only for literacy, yeah, in general, information literacy, linguistic literacy, and the second one is literacy of mathematics, namely numeracy, yeah. But for the teacher in the future, for the people in the future who will survive in the 21st era, it is not enough to have a basic literacy. A basic literacy, I already mentioned about science, social science, and everything, because we are now in the internet era, of thing era, in the disruptive technology era. We need to have another higher literacy that beyond of those literacy. That's we call meta literacy. So we already asked my student as well to to do research about this one, especially in postgraduate student. We already find out with the completed the instrument of the meta literacy. Yeah. So meta literacy actually based on the information literacy right now, maybe we can say that, okay, we already enough to use, we know how to use a computer, we know how to use internet and everything. But what happened without having a meta literacy, sometimes student only, or if we given a student a task or assignment, and then they ask to Google, and they ask to Google Classroom, Google Scholar, they ask to YouTube, they know YouTube, and finally what? They just copy and paste. So because this thing, because they are only having information literacy without any analyzing, without any incorporating, without any evaluating. So they just copy and paste, and finally they give to the teacher, they give to the lecturer. That is my assignments or result and already. And finally, if we check the Turnitin checker similarity, there are a lot of similarity. So the humanities literacy and the meta literacy also a main concern about this. Yeah, thank you.
Thank you. So, if I may elaborate more, Prof, it is also stated in Mr. Ahmad Jainuri question that meta literacy is not the answer of our national problem because meta literacy is not reflect on the value of Pancasila. What do you think about this, Prof?
Yeah. So I'm thinking about the skills and literacy is a bit different. Yeah. If you are talking about skill, for example, HOTS, higher order thinking skill, yeah, starting from analyzing, evaluating, and creating and everything. And literacy in other sides, we are talking about literacy. Literacy is the minimum requirement for people who live in this era, for example, living in the situation that we have something. But in meta literacy, in fact, even we consider about humanities. The humanities not only working on the computer base, also mobile phone base and everything, but also thinking about the communication and collaboration with each other. So when you interact with each other, you should develop the character, that Indonesian character, of course. You should respect each other. And also you should share something which is you now deeply about this information. So first, you need to characterize whether this information right or not. You need first to characterize whether this information true or not. I believe this is the reflection of the Pancasila, our indicator itself. So I believe in the meta literacy, if you just have information literacy only, it is that you are worried about. By completing with the meta literacy, that students should be awareness about the humanity literacy, not only data literacy, technology literacy. Data and technology literacy, yes, sometimes will lead people, yeah, selfish. You will lead people, individualists, for example, they just know about computer, but meta literacy beyond of this, they should consider about the character building as well. So actually, this is in line with the what Pancasila reflection is there. Yeah, thank you.
Thank you,
Should I
Pak.
continue? Should I continue now?
Yes.
Okay. Sorry, a little bit problem with that. Yeah, okay. So again, by having this problem, then what do we have a problem with the meta literacy in Indonesia? Yeah. So actually, we do not finish this study yet, but for the information literacy, yes, we have a problem, especially for the teacher. For the teacher itself here, okay, this is, we done 1 year ago about survey of information literacy. Yeah, yeah, because but the basic of the literacy on meta literacy is what information literacy. The survey result on 200 school teacher of ICT proficiency. This is about the literacy of information. So what happened? Communicate 55%, evaluate 40%, access 60%, and manage and integrate only 15%. The creating level, creating activities by using Internet of Things, also by using the platform and everything, is very less among the teachers. Among students, it's a little bit bigger, but it's not so big, yeah, compared with other. The biggest indicators along with the surface, so when people can access internet, accessible to internet is yes, we are no doubt about this one. Accessing internet all the time, accessing gadget all the time, communicate with internet all the time. But when they ask to create something through internet, it is a big problem. Yeah, for Indonesian teacher especially. So we have a problem here. So that's why need to have research about this one. Why we have a problem with along with this survey on the information technology literacy? Because in Indonesians, I believe there are already people now about this, and most Indonesian teachers are in the age of baby boomer. So if we have a millennial age 18-29, that's our student, yeah, our higher student, of course, yeah, for a school student, even better, maybe not millennial, maybe Z generation or something like that. and also teacher with what generation X, like age of 30, 45, baby boomer. So this one, I believe most of the Indonesian teacher in this stage, 46 until 64. This is the also. So that's why our research problem now, how is the syntax of model integration, yeah, by using RBL model into STEM education? This is the first research problem. So we integrate research-based learning model into a STEM education to improve the student meta literacy. The second one is how do we obtain? So this is the another problem, the rainbow anti-magic connection number. And the third one, how do we use the rainbow connection number for developing a secure crypto system? And finally, can the RBL STEM, yeah, research-based learning STEM approach improve the student meta literacy in developing secure crypto system by using a rainbow anti-magic gallery? So this is the ongoing research that we have been done right now. So actually, we have focused on the meta literacy improvement as a student learning outcome by using a research-based learning model and also by combining with the STEM education. So research-based learning, I believe we already all the time presenting about this one in front of our lecturer and also student. Firstly, research-based learning, written by Marzano and Pickering, Pollock 2001 about classroom interaction network, yeah, namely research-based strategy for increasing student achievement. So, and there are a lot of Alan Jenskin finally also following by Marzano books, and also Alan Jenskin and Michael also together. So what is in the research-based learning? I also mention all the time about this one, and I already implement think this one also in the classroom. There are three elements that should be avoided, shouldn't be avoided. The first one is lecturer, and also class and research group. So I believe some people already know about this one. Yeah. So research group, what is the research group? I believe also this one already aware, but people already know also about these ones. In some other university maybe has not been implemented. It's not been implemented, but in our faculty, in this university, the research group in the department level, faculty level, or university level. So the main key of the research group, student research, is also our lecturer research. I believe we have also this one, yeah. So research-based learning is also actually part of the existence of the research group. If the research group can develop topics and also research group bring that topic into a classroom, into a research activity, actually this is the philosophy of the research-based learning. Yeah. So people talking about research-based learning, if they are teacher, they bring their knowledge about research in daily activity and their research activity into a classroom. That's the research-based learning. So you can bring the other scientists' research into a classroom as well, but it's not enough. The research-based learning should bring their lecturer, their teacher, and every people who are teaching in the classroom, they bring their research in the classroom. So Alan Jenkins, McHaley together, 2007, gave a four diagram of research-based learning about the integration of research-based learning. So in the quadrant of this research-based learning, the fourth quadrant is actually the involvement of learning process actively involves students to do a research in solving a problem arising from energy problem. And finally, so this one also the difference between research and discovery inquiry-based learning. Discovery or inquiry-based learning and research-based learning. What is the difference? Actually, the main difference in the research level. In the research level, For discovery or inquiry-based learning, it's not necessary to be new, but in research-based should be new, even a simple one. So which is a part of the novelty. So we call this novelty actually, even in the classroom. So we should part. So this syntax of phase of research-based learning, we already published this one in the International Journal of Instruction. Also, we have another journal publication about this syntax. The first one is there are 6 syntax in the research-based learning. Problem posing rising from RG, open problem. Students are encouraged to obtain a breakthrough. And then orientation data collection, tabulating hypothesis, data analysis, predicting process, validation, investigation, analysis, generating, testing, conjecturing, creative new concept, and also the last one, FFGD. This is the syntax of the research-based learning. Now we are talking about STEM. I don't want to go into detail about this one. STEM already known. STEM is education approach, STEM approach in the classroom. If teachers involve science, technology, engineering, and art, mathematics when they are teaching in the classroom, actually they use a STEM education already. So STEM education acronym was introduced in 2001 by scientific administrator at the U.S. National Science Foundation. And STEM education, actually the curriculum construction should be based on the idea of educating students in 5 specific disciplines: science, technology, engineering, art, and mathematics. So that's why STEM education is interdisciplinary concept of education. So there are 3 things of science STEM education. There are 3 types of STEM education in the curriculum. The silo approach. Yeah, the silo approach, you teach a student separately. You teach science and art, you teach technology and engineering, you teach mathematics separately. But if in the embedded approach, when you are talking, for example, mathematics, in the mathematics, you are talking about real life, real life problem. And then you need technology and engineering. You involve technology and engineering. You involve in science to solve that problem. But the nice one, STEM education, the integrated approach. What is the integrated approach? It is teaching and learning what model based on the contextual teaching and learning model. So there is real life problem, and the real life problem should be considered whether they need science, technology, and engineering or mathematics to solve it. So this is the STEM education. So normally source of the STEM problem based on the SDGs element. SDGs element, you can have a no poverty problem. There are a lot of our poverty problem in Indonesia. Yeah, also there are a lot of hunger in Indonesia. So the SDGs program gave the 17 indicators. Zero hunger program is makes Indonesian people, the world people, free of hunger. Also good health and well-being, quality of education. So there are a lot of problems there. If you bring this problem into education into a classroom, then you have a STEM. So the STEM look like in the classroom, students are actively having a simulation, students are actively having outdoor learning, for example, plantation and observation. Then students are very actively interact with the atmosphere and the environment. Yeah, either this is in the garden or everything. Yeah, okay. Student also do something. Yeah, this is outdoor laboratorium. Even also in Indonesia, we have educational edu farm classroom. For example, yeah. So students are asked to join excursion by a lecture teacher and go to the edu farming classroom. So this is so I believe in the level of student memorization and student participation in the classroom for STEM education reaching this stage. So In the classroom, if you just ask students to read, to hear a word, looking a picture, watching video, looking at the exhibition, it's not enough. So the students should be motivated to do a simulating the real experience, doing the real thing. That is the good one. So what is the memorization? 90% of memorization. And this is active, learning by doing. Yeah. So research method in here, I believe the current research method is in general something like mix of the developmental research, and also the quantitative research. We have also research finding along with this research in our study. We have already published a paper about RBL and for metacognition informant, 2019, RBL for student achievement improvement 2019 as well. Also, Rido et al. RBL for creative thinking skill improvement. So we already published this one. So you can have look. Even we have also very recent publication of RBL. But for a combination of RBL and STEM, we do not have a publication yet. Yeah. So this was the syntax of integration of RBL model into STEM education. This is what this answered the first problem. In the first problem, what is the syntax of integration RBL model into STEM education? So the first one, we have a look when we mentioned in the research-based learning syntax, and then we bring the research-based learning syntax and integrating into STEM education. And then we have science, we have engineering here, we have technology here, we have engineering, we have mathematics, and we have a report. It is very nice, actually. Yeah. So one, we have engineering. Engineering actually not necessarily related to technology. Engineering is just some techniques. So a breakthrough, new breakthrough, for example, to have to solve a problem. In here, a breakthrough related to encryption process in online transaction data and collecting data using a technology again. It's developing a secure crypto by using a rainbow anti-magic. This is engineering and analyzing the rainbow anti-magic color. It is in the classroom. So the students are encouraged to use a STEM, science problem, technology, engineering, and mathematics. This is the STEM, but the syntax follows the research-based learning. So we have a combination of research-based learning and STEM. So what is the STEM element? In the STEM element, according to Margaret Karparu, so we have also the involvement of the science, technology, engineering, and mathematics, because we use the integrated approach in the STEM. So we use all of them: science, technology, engineering, mathematics. When we have a problem in the e-commerce security system, is the most crucial issue actually in online transaction. In general, the e-commerce security system require security protocol to encrypt and decrypt crypto key on the web server. How to develop a security system, a secure crypto system, about encryption and decryption is a big challenge. So we couldn't solve this problem by using only mathematics. We couldn't solve this problem by using only engineering. So it is complex, actually. So the elements of the STEM should be involved to solve this problem. So this is the integration of research-based learning into a STEM. Then finally, we have this one. So for example, so this one, the first, in the top, but we have a problem. And we use a rainbow connection and anti-magic coloring. So to have a crypto system, to build encryption and decryption. So how does it work? For example, here we have the rainbow anti-magic coloring of graph. I don't want to go detail these concepts, for example. And then we have a illustration of the rainbow anti-magic coloring. For example, we have a cycle here. And then we found the cycle, we assign label into second such that the H weight the two adjacent vertices having a different edge width. So this is anti-magic labeling. But how to bring this one into anti-magic coloring? Yeah, anti-magic coloring not necessarily having all this edge width are different. So consider here 7, 7 are the same, 6, 6 are the same. Yeah, but it is already a rainbow anti-magic coloring. So by using this rainbow anti-magic coloring, for example, I using this kind of network topology, and then we implement this one to the security system. That's we call a crypto system. How to use it, this one? For example, I use this one. Yeah, encryption process, we use YA, YI, XAI plus KA modulo 26. And decryption process, of course, inverse to inverse of the function. Yeah, to inverse the function again from plain text and to a ciphertext and ciphertext into a plain text again. So how do this would work? For example, we have a plain text. Yeah, if you have a name, of course, it's okay. FKIP UNED. By using this word, by using the rainbow anti-magic codes, and then we have 1, 2, 3, 4, 5, 6, for example, and then I put this one in XI. XI 5, 10, 8, 15, 20, 13, 4, 9, yeah, which, where does it come from? Okay, it come from this label, yeah, and then we use a cipher cheaper to convert plain text into ciphertext. So if you input your name, for example, in the online transaction, your name, for example, David, then how the computer encrypt that name in such a way it is not revealed by unauthorized people, they should pass the crypto system. And how do we generate encryption? Now we use rainbow anti-magic coloring. We train this one to our student. In the classroom, we bring this one to our student, and student working with the different data input, for example, different number, for example, and we analyze their meta literacy. So FKIP Universitas Jember, by means of the rainbow anti-magic coloring, FKIP Universitas Jember as a plain text, and final the ciphertext we have gone rust. I believe people couldn't read this one. What is that? What is that? Because this is already encrypts. If people, the recipient will read about the encryption, again they should decrypt and they should go to the inverse of the cipher. So this one that I've done now, this one that I've been doing in the classroom. So we have this tool, and then we implement in the classroom, and with the special indicator, special main indicator of the meta literacy, we also develop a instrument of the meta literacy. Finally, we get this kind of different. And fantastically, the student in the pretest, yeah, before implementing the research-based learning and STEM education, they just average 40 out of 52. After this one, and then I just testing. This is just a simple and straightforward experiment, our non-experimental research. This is just testing a pre-test and a post-test, and finally they got higher, the meta literacy higher than the previously in the pre-test. So I believe it is a potential for a student to encourage their learner to have a meta literacy by implementing research-based learning in STEM. So if you already only implement the research-based learning, project-based learning, for example, discovery-based learning, it's not enough to improve their meta literacy, because they couldn't know about the real world problem. And they do not know how to inform about the science, technology, and engineering. Maybe they just inform engineering. Maybe they just inform mathematics. What about the technology? So all the time, if you use STEM, I believe the TPCK, technological, pedagogical, content knowledge, will be involved all the time. And that time, the student or the learner meta literacy can be achieved by this kind of situation. So I believe as the concluding the remarks that the implementation of research-based learning as well as a STEM approach can improve the student meta literacy in developing a secure crypto system by means of rainbow anti-magic coloring. This is in my case, of course, a various type of graph representation. Student meta literacy portrait fish indicate how differ their individual thinking skill to develop a secure crypto system by means of rainbow anti-magic coloring and determine the student meta literacy under the implementation of research-based learning and STEM approach. And finally, we have maybe in the future for the open problem, we need to develop a GUI, graphical user interface, with a specific programming on the implementation of rainbow anti-magic. In such a way, the student meta literacy will be increased one time to time, and this is actually the what open problem for the future research. So I believe maybe we can continue having a discussion, Ibu Siti Mordia. That's the end of my talk now, and then just wait for the question. Thank you very much.
Thank you very much, Prof. Please, ladies and gentlemen, give warm applause for Prof. David. So now with that, we now enter to the second segment of this session, which is question and answer session. So some moment ago, Prof, we have one participant who raised hand. Maybe, if I'm not mistaken, Miss Pak Wahyu.
Yeah, okay.
Pak Wahyu?
How are you, Pak Wahyu?
Pak Wahyu, are you there, Pak Wahyu?
Okay.
Pak Wahyu.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh, Pak David.
Waalaikumsalam warahmatullah. Associate Professor.
Thank you so much, Bu Murdia. You give me a chance to deliver questions. Very wonderful topic, Pak David. I think meta literacy is very, very important to, especially for the teacher, especially for higher education, because meta literacy is one big indicator, the performance for teacher to handle the class, and also a big performance to the student, our students. Even meta literacy also strategic performance for the nation competitiveness according to this era. Now, my question is very simple and pragmatic questions. How to implement to the teaching programs where from we start? What should be prepared by a teacher before implement the meta literacy teachings, and what aspects should be concerned for the teacher?
Yeah, thank you.
Thank you.
Can I ask Ibu Murdia? Can I answer? Yeah, okay.
Yes, Prof. Yes, Prof.
Yes, very nice, Dr. Wahyu. Long time not to see you, and I believe and I wish you also stay healthy.
Amen.
Every people also here. So as I mentioned in the main indicators of meta literacy, the first one is produce. Second one incorporate. Also use, share, and collaborate. Apart from those, in the meta literacy should have and should put a big concern about humanity literacy, as well as should comply about 4 domain of competency: cognitive, metacognitive, behavioral, and affection. So this is part of the learning outcome, just like thinking skill or everything. So to measure about the meta literacy, to measure about the learning outcome, the lecturer or the student, the teacher should develop instrument of test, of course, either using writing test or speaking test, observation, and everything. So because this is about learning outcome. So teaching material should be performed first, should be prepared first by a lecturer, by a teacher, if they want to measure about their student meta literacy. But it's not enough. So when we go to a classroom, we just bring an instrument of test, instrument of observation, and ask a student to do something, and distribute a test. It is not enough. We need to have a model. The model in our study, namely research-based learning. Research-based learning is a model of teaching and learning. Of course, we know the syntax of the research-based learning already. We have already publication in the Journal of Instruction, yeah, already have a publication, the UJR as well, Universal Journal of Educational Research about research-based learning. But the question, does the research-based learning support the existence of meta literacy? And I have done a research about this one. The answer, no, because research-based learning only asks students to do research, even in the laboratorium, even in the other, in front of classroom, and also doing something in the classroom, in the outdoor classroom, for example, which is not having involvement of technology, which is not having involvement of information digital devices. So that's why we need a STEM, science, technology, and engineering and mathematics. So in this study, we try to integrate, we try to combine research-based learning and STEM education together in order to build or to improve their meta literacy, the student meta literacy. Of course, when we already determine the research-based learning integration into STEM education, so what do we need now? As a lecturer, as a teacher, we need to develop a syntax of the integration of that model and approach by having the syntax, and then finally we should develop a teaching material. Yeah, of course, in the university level, yeah, for example, semester teaching plan, we should develop also the the others, for example, student worksheet we should develop, and student assignment we should develop, and everything we should prepare. That's one. We already asked my student as well, especially in the postgraduate student, yeah, already developed about the teaching material, and also they already implemented. So this one, the first step, the step to implement the research-based model integrated with the STEM education to improve the meta literacy. So I believe in the syntax that I already mentioned, it is quite clear how the teacher or the lecturer can implement the RBL together with STEM education to improve the meta literacy. My answer is clear. Sorry about that. Yeah, I give it back to moderator.
So, any feedback maybe, Pak Wahyu? Halo Pak Wahyu. think it's a technical
Yes,
problem.
maybe he lose the connection. Yeah,
Yes.
I saw his picture is not moving.
So
Okay.
I think we can move to the next question if you may, Prof.
Yes, yes,
While
definitely,
waiting for Mr. Wahyu.
definitely, definitely.
Okay. So, well, actually, this is the continuation of previous question from Mr. Ahmad Zainuri. Apparently, that he is still uh, not satisfied with your answer, Prof.
Yeah, no problem.
About how metal literacy actually cannot solve the problem because it does not reflect the value of Pancasila, sir. So
Yeah.
let me read the question, the second question from Mr. Ahmad Zainuri. Well, metal literacy is oriented uh, towards the cognitive only. So how come that metal literacy can be reflected on the Pancasila value? Because Pancasila value mainly concern about soft skill development, not cognitive development, sir.
Yeah. Okay. Actually, I already answered the what the question, but okay, I just repeat again. In this what domain of uh, meta literacy, so you can have a look. Meta literate learner. That's a Jacobson and McKay said in that in their book. Yeah. So of course there is a cognitive, behavioral, affective, and metacognitive. In the inner layer, in this cycle, there is what competency. So very student what graduation competency here should comply about cognitive, behavioral, affective, and metacognitive. So when you talk about this literacy does not reflect a Pancasila, what for example character or behavior or something. So might be this regard to information literacy. But in meta literacy, beyond of information literacy, they the learners having a meta literacy should pass the 4 competency domain. And the second layer from inner layer, look at that. Civic minded. I believe it is a strong message for the civic minded is humanity. Humanity, I believe it is a reflection of Pancasila value. Yeah. So one is that telling us, even you are not in a classroom, you are not facing your student, but you're facing, for example, by Zoom, yeah, cloud meeting. At least you should, yeah, say hello to your student. Not all the time you're talking. Student, what mute their video, mute their audio. You do not care. It is not like reflective teacher to build up their student behavior. In the meta literacy, even you use gadget technology, mobile phone technology, and everything. Yeah, you need to have interaction with the student, with the learner. So this is important. When I am teaching in the classroom, I always say, okay, now time to unmute your video, just like photo session, for example. Need to unmute, yeah, not all the time. Yeah, but there is a reason, of course, for having position video on. It is getting exhausting student all the time. Yeah, to have this one. And participatory, collaborative, and productive, adaptability. This is reflection of the Pancasila value, of course. Yeah. And the outer layer, for example, here, the production, PKIA, productive, creative, innovative, and affection. Collaborator, producer, publisher, researcher, participant, communicator, translator, and author. So this complete what indicators apart from the information literacy. So the completion of this one, I can tell you about this one. In the meta literacy, they have 4 competence of domain. Yeah, metacognitive. Yeah, metacognitive actually in the top level of the logic, analytic, creative, metacognitive. This is in the top one. Yeah, and meta literacy, top of the top. Meta literacy above of the metacognitive. But cognitive is a different side. They are talking about Bloom taxonomy. Yeah, from knowing, remembering, understanding, analyzing, evaluating, creative. Above that one in metacognitive. But in the literacy, it's different side. Literacy is scientific literacy, mathematical literacy, final literacy, sign literacy, and also information literacy. Above that, the top one is meta literacy. It is different side. But if you are asking about the completeness of meta literacy indicator with the fulfill of reflecting Pancasila indicators, yes. The answer is yes, because we have 4 domain of competence. Thank you very much.
Thank you, sir. I think it's elaborate enough, and I hope it's already answered Mr. Ahmad Zainuri's question. So we will still have a couple of minutes actually. We will end this occasion at 11:30. We still have 6 minutes. So I will take maybe one more question from the audience. Is there any? Maybe you can raise your hand, please. No. All right. So there's no more question, Prof.
Give concluding remarks before.
Yes, Prof. Please, please.
There is a chart here. Maybe someone asked. Okay, now.
No, there's no.
Okay, participants, I already talk about 4 things. Yeah, or in general, we have talked about 3 things: the model, integrated by approach, namely research-based learning and STEM education. STEM is approach, not a model. Research-based learning is a model. And also, we have a learning outcome to achieve, whether literacy or skills. Skill is part of cognitive, literacy is different side. And as well as a problem. Problem could be from everyday, real life problem, daily life problem, could be taken by social life problem, from daily life problem, or by having a problem which is in the context of the science, pure science, or anything. That's a complete research of interest in the doing a research. If we only develop a research-based research model, for example, our teaching and learning model, or develop an approach, we do not care about the learning outcome, it is also hopeless, because some model and approach already established, already published by some other people from all over the world. You can have a look in the top journal, which is indexed by Scopus or WAS. There are a lot of models that's already developed. But if you're talking about only learning outcome, the thinking skill, all the time you are talking about creative thinking skill, critical thinking skill, all the time repeatedly you ask students to do this research, which is already published by our people. I don't care, you can have a novelty of the research. I don't care, you will have a good research. So all the time you have talk about creative, innovative, and also collaborative and everything. So I'm thinking that if I brush the creative thinking skill, which is a topic taken by a student, and I browse in the repository of the University of Jember or another university, there are thousands items found. Yeah, so it will be worthless. But how do you have a good research? You should talk about the content as well. Don't avoid the content. A content is a very wild problem because content developed by some other people. So content together with research-based learning or research learning model together with approach, and then integrating in the others technique, for example, this is actually the potential novelty of your research will be reached in the future. So please involve these three of things at least: model or approach, learning outcome, and then finally the content problem. If you're talking about content only, sometimes the implementation, the application of this content, it's not so rich. But if you're talking about the model only, sometimes it is just a repetition of the model that people others already taken for their research. Yeah, even also for the learning outcome. So please do not do research in which the answer already known before doing a research. The good research, if you start research and you get confused before starting the research, this is a good research. If you know already the result, the answer, it is not a problem to be a research for the future. Good luck. Yeah, having a good research in such a way you can have a good publication for the future career, for the future promotion, or for finishing your PhD program or master program. Thank you very much, Ibu Murdia. Thank you again. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Prof. David. Ladies and let's give Prof. David a round of applause. So we've come to the end of our first round presentation, and we would like to thank everyone who make this occasion possible. And I thank all of our presenters, Dr. Ramesh and Prof. David, for the valuable presentation, and for all participants who coming and listening. So thank you very much. I'll get the mic back to our MCs. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much.
Ladies and gentlemen, the second plenary speaker session, we will have Dr. Murti Pichumani. This session will be led by Mrs. Robiatul Adawiyah, SPD MSI, as the moderator. We have 1 hour for the second plenary session. Moderator, time is yours. Check.
Ya, Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to the second session of plenary session, the third International Conference on Physics and Mathematics for Biological Science, I am Robiatul Adawiyah as your moderator in this session. Okay, well, ladies and gentlemen, in this session, we will have one invited speaker from Department of Nanoscience and Technology, Sri Ramakrishna Engineering College, Dr. Murthy Pichumeni. Hello, Dr. Murthy. Can
Very
you
good.
hear me?
Very good morning. Yes, I can
Okay.
hear you.
Okay, how are you?
I'm fine. How are you?
I'm fine. Thank you. Okay. While waiting for Dr. Murthy Pichumeni preparing his presentation, maybe I will read his short curriculum vitae. Okay. Dr. Murthy Pichumeni is an associate professor in the Department of Nanoscience and Technology, Sri Ramakrishna Engineering College, Coimbatore, India. And he was born on 26th of October 1985 in Tamil Nadu, India. He got his Master of Science degree from PSG College of Technology, Anna University, Coimbatore, India. And he got his PhD degree from University of Navarra, Pamplona, Spain, with his research work, Effect of External Fields on Dynamic of Colloidal Phase Transition. And his expertise is in the nanoscience and nanotechnology, especially in the nanomaterials, nanofluids, material science, soft matters. Okay, well, ladies and gentlemen, similar with the previous session, we have a role here. We have about 1 hour for presentation and also discussion. Maybe about Dr. Murthy, you have about 45 minutes, and the last 15 minutes we will have a discussion. If you have any question, you can write in the chat box, or if you want to ask directly to the Dr. Murthy, so you can use the raise hand features in the Zoom meetings, and then I will call your name. After that, you can ask to the Dr. Murthy directly. Okay, well, Dr. Murthy Pichumeni, are you ready?
Yes.
Okay. For Dr. Murthy Pichumeni from Sri Ramakrishna Engineering College, time is yours.
Thank you very much. Good morning, the participants. Good morning, delegates. Namaste from India and salam. I wish each one of you a very in Indonesian time and good Indian time. It's a very good opportunity for me to have this interaction with you, with all the delegates from the university. It's really feel very happy that the University of Jember is doing very well in this kind of, and I'm very see that third edition that this organization is conference in line. Very good congratulations to one of you. I would like to share the presentation that I have with me. Dear organizers and participants, can you able to see the slide clearly?
Yes, yes, doctor, we can see your slide.
Thank you. Thank you. The short abstract that I would like to present here for this international conference on physics and mathematics for biological. The theme that I've chosen to share with all of you is small is different. So if I say the word small, I am going to directly to the word called nanoscience and technology. Small, it goes explaining the terminology called nanoscience and technology, and nanoscience and technology, how it is going to be different, how nanoscience and technology is going to be beautiful, science and technology is going to be very interesting. So this is the theme of presentation that I've taken today. And during the first half of the presentation, I will introduce a few fundamentals associated with nanoscience and technology. And in the second part, I would like to share the applications of nanoscience and technology, that is a small and this presentation to correlate with the very high theme that Professor Dr. Dafik presented earlier, that is science, technology, engineering and mathematics, STEM, that is one of the core that global is moving. So it is a it is it's a merging of four different domains: science, technology, engineering and mathematics. As Professor Dafik rightly pointed, to work on real life problems so that we can get good publications and the research quality will be very, very high. Like STEM, this nanoscience and technology is also a transdisciplinary and interdisciplinary domain. Usually with this lecture, I start with a very interesting word that for the other domains like mechanical engineering, if I need to say a mechanical engineering or a biological science or a chemical science, they are core domain. Whereas nanoscience and technology, it is a interdisciplinary and it is a transdisciplinary domains wherein experts from different domains, for example, from chemistry, from electrical engineer, from biology, they join hands together in developing a product. Okay, so this is one interesting scenario with the nanoscience and technology, that is a transdisciplinary and interdisciplinary domain. Let me navigate further. If we imagine that if we are constructing a building or if we are constructing a home, so starting from the soil analysis, a geologist can do. Later on, architect can go for the design, and engineer who is expertise with the civil structures will do the building. Then we have expertise from electrician, plumber who makes very, very interesting paintings, and the interior decorator. So just to have a construction being in the place, please see that there are different experts are joined. Am I right? Do you agree with me? Starting from the geologist, architect, engineer, electrician, plumber, interior designer who makes designs, painter, or carpenter who works with the woods and so on. So just to have a building, we have a lot of domains being involved in such a way that this nanoscience and technology which contributes for the STEM is also involved with the domain. Can happen in the our roadsides. Probably this is a scenario where our police friend would help the society. For example, if there is any people who consume the alcohol beyond the limit, to check. For example, in Europe, they have these kind of devices in place. Consumption or not, which is very bad for the health, very bad for the society. So when you look at it, they have a, can you able to see my mouse pointer moving? They have a small device holding in a hand. Actually, the name of this device is a breath analyzer, or commercially it is called as an alcohol sensor. When you breathe, the sensor senses the alcohol molecules and gives a output in this display. So who can able to who can able to collaborate and have this device in place? For example, a material scientist, a biological engineer, electrician, electrical and electronic engineering, E&I, electrical and electronics instrumentation. And in order to do a batch manufacturing, we need a mechanical engineer. So just imagine starting from a electronic domain and a mechanical engineer, moving on, so many number of domains are being involved in developing this product. This is the main thing that the first take home message that I would like to give to all our participants, that this is a nanotech science and technology. It is a transdisciplinary domain. So this is a very, very first take home message, that the collaboration from science, technology, engineering and mathematics is need of the hour. Coming back, So what is the very actual length scale that I'm talking about? Okay, so probably you know very well that the nano science and technology, or the small, is dealing with the 10 power minus 10 or 10 power minus 9 meters, comparing with the other objects. For example, can you able to see my pen holding in my hand? So this is the length scale close to 10 centimeters, and this is the cap of the pen. This is close to 4 centimeters. So these objects can be seen very clearly from our naked eye. And nano objects, they require a special tools, and the length scale is of the order of, for example, 10 power minus 9 to 10 power minus 10. It is very difficult to give a realistic feeling of how this nano science or nano materials would be. For example, I can give another example. We have a human hair. This the diameter of the human hair, this is close to 100 micrometer. Now imagine you take a single strand of hair, and you try to cut into 10,000 times. I mean, not like a carrot cutting, it is like cutting in a transverse side. You try to cut the hair into 10,000 times, we will end up in a nanoscale region. So this is a kind of estimation that we can able to project. And why these nanomaterials or nanoscience and technology that are being playing a huge role in the market? I've taken very interesting examples. A lot of cosmetics available in our market, cosmetic products available in European and in Asian countries. For example, a bathing soap that contains nano silver particles, and a cream that contain nano activated carbon. There are some paint that can able to purify, since they have activated carbon technology. They absorb the moisture, they absorb the dirt particles from the air. There are some other products available in the, it's a, I mean, whenever whatever we see in the market, these kind of cosmetic products or products associated with the electronics, there are at least a nano material being involved, or at least chemicals being involved. And when we go into the real force, the driving force that is really needed for this 21st century, to for the better research kind of a habit that to develop, it is a four driving force that are available. The concept is intelligent, fast, multifunctional, and low cost. Nowadays we are using a lot of Android phones. Remember before 2000 or 2005, we have an older version of mobile phones where we can send messages, we can call, and we can do very limited activities. With the Android phones that we have with us, we can able to do a lot of tasks. I hope you agree. We navigate using maps. We have a phone. We do a video recording. Am I right? We do a audio recording. We used to study. We used to attend events, conferences. We have a multimedia activity. So it is very intelligent. The Android phones are very intelligent, and it is robust. For example, with the fingerprint or with your face unlock, the entire mobile phone, it works, and it is multifunctional, as I have given a various lot of examples. And it is also low cost. For example, US dollars of 20 or 25 US dollars, we can able to get a Android phone, or even with the with the 50 US dollars, we can able to get another high-end mobile phones, Android mobile phones. So the main concept with the industrial technology, along with the science, technology, engineering, and mathematics, towards the research purpose, is the concept of miniaturization. So this is a second take-home message that I share to the researchers and the students of this international conference. The first one is transdisciplinary. Second one is the concept of miniaturization available with the research, in particular with the industry. There is a gap between the industry and the academia. So industrial requirements will be on the upper order, whereas the academia research is a bit low. I mean, it has to match with the industrial requirement. So we really need to equip to match with the industrial requirements. with the academic performance in research and other research and the development, research, development and demonstration. So it is not only the word research, it is R, D and D, research, development and demonstration, or it is research, development and deployment, RD and D. So in going further, as I've stated that nanoscience and technology, not only with the applications with pollution or with biomedical applications such as healthcare and the sensors used in the hospitals, clinic, biological systems, electronic devices, it also used in agriculture area, because agriculture is very, very important for a country to grow. I hope you all agree with this statement. And also this helps in a major energy conversion and energy storing devices, this nanotechnology. So this is a very good transdisciplinary domain. As I've stated, people from different domains, for example, researchers from biology, researchers from mathematics or applied mathematics, or from researchers from environmental engineering, researchers from, for example, mechanical engineering or computer science engineering, you can use nanotechnology as a tool to solve problem. There are a lot of problems like energy, environment, and medical problems, water scarcity, we have an oil crisis. So we have a lot of problems, and we can take nanotechnology which give new properties, and finally we can end up with the nano. So this is the main reason that nanotechnology can be used as a tool. So this is the third take home message that I would like to share. Nanotechnology is a tool where you can use in your domain to solve realistic problem. So I'm going to take only 1 minute to explain what is the uniqueness of this small, that is nanotechnology. The first thing is that nanomaterials, they possess increased surface area or improved surface to volume ratio. Imagine this is a single cube. When you calculate the surface area for a single cube, it will be totally 6a square. But what I do is that can you able to see here the mouse pointer moving? You try to cut these small cubes into, cut this bigger cube into various smaller cubes, obviously increasing the size, decreasing the size by a by 2. And now the check, when we try to into various smaller cubes, the surface area has become from 6a square to 12a square. So that is a very, very increase in a surface area. If I need to take a realistic examples of how to understand this increase in surface area, we can take the art of cooking. For example, I've taken a here, and with the potato, we usually cut into smaller pieces. We not only these kind of bigger vegetables, you can also take other vegetables like pumpkin. We cut into pieces. So what is the reason for cutting and cooking is we are increasing the surface area. So when we increase the surface area, it would be cooked very well and very faster. And this is another important thing. So this cumulatively will be the fourth take home message, dear participants and students, that there is a change in the properties. For example, if I take a bulk material, or if I take a material of the for example in the mill, and when I change the same material into a nano, or when I change the material to nano domain, there is a change in the property. So for example, this is a bottles filled with colored solutions. It's a very interesting and fascinating colors, but all bottles, they are same gold nanoparticles. The only difference is the size is changed. Changing the size of gold nanoparticles, we can see interesting optical properties which find interesting applications. Also, we have a lot of change in the electrical conductivity. For example, we can take a pencil. We can take a pencil. Can you able to see in the video? And can you able to see the tip of the pencil, which is having a black color, that is nothing but a graphite powder being stacked. So when you check the conductivity pencil, it is a very good conducting material. But the same graphite, when it's transformed into a form of a carbon nanotube, a nanomaterial, this will, there is a change in the electrical change in the optical properties. I proceed various type of nanostructures that we are going. Example, quantum dots. They are nothing but island kind of a structure, and nanowires, nanofilms. They are one-dimensional and two-dimensional structures. Probably we'll be using these words during this presentation: nanowires, nanofilms, nanoparticles, and so on. So they have their interesting applications, and the synthesis of these nanostructures. Synthesis is nothing but the preparation of these nanostructures would take a different. Let me go for a few realistic application. Usually we learn from our nature, or we try to mimic from our nature. I have given here a very wonderful and beautiful butterflies with or a gem crystal, or it is, and this is a desert with a lot of fascinating colors on the surface. How these butterflies, beetles, they get colored. The thing is that each and every surface of these beetles and butterfly, can you able to see here a highly magnified picture? They are having a crystals being arranged periodically, or biocrystals, or they are called as a photonic biocrystals available with these materials. So in the case of semiconductors, semiconducting materials like silicon, germanium, where there is a manipulation of charge carriers, electrons and the holes. Whereas in this case of these materials, there is a manipulation of photons. So that is a reason it is analogous to semiconducting crystals, photonic crystals. Another interesting creature that I have shown here, that is a chameleon. Usually we have a chameleon changes color depending on where it is being placed, but it is not so. This chameleon, when you do an investigation, when you look carefully, this chameleon having the nanocrystals on the surface. Chameleon also, they are very good pet animals, and these chameleon, they have the in the surface they have a nanocrystal, okay, that reflect pigment. So if the chameleon is very hungry, it will change the color, and if the chameleon is very angry, it will change the color. Or if chameleon is finding some threat from the other animals, it will change color. So chameleon, it changes color depending on the mindset. It is not depending on taste. So it reflects different colors. Not only the chameleon that we usually forest in the park, they also exhibit these colorful skin. Actually, these gecko lizards, they have a nanocrystal on the surface of the skin, and they can able to vary. Can you see this ordered nanocrystal, a smaller crystal? These are nothing but photonic crystals. Can you able to see this research going on in this in the nanocrystal of these small creatures? So these are photonic crystals, a very good biological application wherein we understand, correlate the nano with the biological application. So when we look into this, we can also correlate the photonic crystals with the physics. For example, in order to a photonic band gap like a semiconducting band gap, a loss from the Faraday ampere, and we need to develop a quantum mechanical approach like a Schrodinger like wave equation. Team is doing research in photonic materials, photonic crystals. There is a domain of physics, domain of mathematical physics, a domain of biology, a domain of engineering from biotechnology, a domain of another engineering from the nanoscience and technology. So this is a thing that I've been keeping on repeating. So here is a integration in the sense it is a vertical. It is not the vertical integration. It is a horizontal integration of mathematics and engineering, because these photonic crystals, they find a very huge applications in the market. Probably in the year 2025 2030, they find a lot of applications in the market, as I have indicated here. Another example that to discuss is the very good flower that we see in the lakes is a lotus. This lotus leaf, one interesting character is place a small try to roll over. The reason is that this lotus leaf having the surface very high roughness. So what happens? Due to the very high roughness, the super hydrophobicity would come in. How we can get the understanding from the super hydrophobicity to a real life self-cleaning surfaces. Imagine that if we have a building of 2 or 3 stories outside, we can able to clean it very well. Maybe manpower with the tube filled with water, we can able to clean. Imagine if we have a building of 40 or 50 stories, it is very difficult to use manpower. If we have a super hydrophobic coating on the surface, these are self-cleaning surfaces. That means if the water droplet flow, when it make a roll, it also take the dirt particle along with that. This is a super hydrophobic nature, and this is a this is a hydrophilic nature. What we copy from the lotus leaf is this super hydrophobicity. If we develop a surface and if the water rolls like this, it can take a dirt along with that. And here also the mathematics being involved. Another important application of nanoscience and technology is with the development of automobiles. Probably this is not with the main scope of the conference, but I would like to say a few seconds of that. For example, we have a crash sensor, we have a inertia sensor airbag, which is a smallest sensor being placed in the car. So what happens is that if we make unfortunately, if we make an accident, this crash sensor, it identifies the force that acts opposite to the direction of the acceleration. Then it gains a momentum and the airbag it activates. So actually the sensor's fundamental principle is associated with the force, momentum, and the acceleration. Here comes the concept of the physics, which is being integrated along with the nanoscience and technology and being given for the another interesting domain, probably from 2020 to 2025, the years that the world electronic industry would be working on is with the smart and flexible electronics. The main cores of the system associated are the sensing element and transduction unit, and obviously a smaller power supply, which could able to give a constant power supply over a wide period of time. And in this kind of sensors, the one thing is the smart sensor. Usually in the olden times, there are sensors where it is not a smart sensor because the feedback that connects with the output and correlate with the input was lacking. However, nowadays we have a new development of a sensor where the partial output is given as a feedback to the input. There it can able to measure very well, and then it is a smart sensor. We can able to see a wide range of smart sensors available in the market. For example, biosensors or biomedical sensors used for the biological application. And if I need to repeat it once again, we have material scientists, engineers from chemistry, mechanical engineer for batch manufacturing, biomedical engineer. They have been taking a very good role in development of the biomedical sensor. Maybe in the next slide, I would like to give a real-time example of that. If we have a chemical sensors, optical sensors, thermal and pressure sensors being used for a wide range of applications. One of the interesting domain that is coming up, probably there are, if students, if you're really interested, the world market would be focusing on two domains in the future. The first one is energy, and second one is healthcare. In the case of healthcare, biology, biological systems, medical devices, medical tools, they are going to take a major strand in this second one, that is healthcare. We have a traditional laboratory analysis. For example, if we go to hospital, if we give a blood, they will take, for example, 10 ml of blood, and they do analysis. Nowadays, there are a lot of instantaneous analysis have come into the market. For example, on-chip analysis, or it is called as a lab on a chip. It is LOC, lab on a chip analysis. It is something like a device. Can you able to see in the video that I'm holding a slide changer? This kind of lab on a chip, the size is something like this. So if we give a microliter of the blood here, it detects and it gives a value. So for example, this entire lab can be integrated in a very, very smaller place. So this is one demonstration that I can able to share here. that we have a blood analysis device. We give a small volume of blood, it reacts, and finally, due to the generation of the hydrogen ion, it can able to quantify what is the blood sample being present inside. I would like to share a very interesting clip associated with that. I'm opening the video for sharing with yes, can you able to see the video? I
Yes.
suppose yes. Thank you very much. So this is a small device that I've said a moment ago, which is having an inlet, a nanostructure for selection of the materials. We have a reaction chamber and we have a deduction chamber. So what happens is that, so we are not giving too much of blood here. We are just giving a microliter of blood here. This device is to sense the blood glucose diabetes disease. So for example, this is a nanostructure which completely removes the biological entities that are not responsible to measure the diabetes. So it only allows the few entities that are responsible for glucose identification. This is a color mapping. For example, green binds with green and blue, and this is a reaction chamber. Then it goes and bind with the reaction chamber, for example, green with green, blue with blue, and finally it goes with the reaction and the values that can be seen very well. So this is one of the interesting application of the biomedical tools and biomedical. Forward, there are in constructing nano robots and the smart can enable a targeted drug. Usually we take in the form of tablets. This goes to the entire body, and if I have a pain in my hand, this kind of nano robots, it goes directly to the place, give the to that appropriate place, and it delivers. Maybe if I have time in the later part, I will show you a clip on that. I would like to move a little bit forward. When coming to the two areas, out of two areas, one area is energy, wherein these nanomaterials, they play a huge role. As I've stated, apart from energy and healthcare, we have a demand for potable water, good water, oxygen. You know that with the pandemic throughout the world is facing a lot of crisis with the oxygen, and we have a crisis with the food throughout the world. So a lot of research, in particular, science, technology, engineering, and mathematics is really required to find solutions in this domain. And nanomaterials, nanoscience and technology, with the help of materials, chemistry, and physics, a lot of energy solutions for the non-renewable, for the renewable energy sources, for solar cells, hydrogen storage, fuel cells, harnessing energy, and tidal in the research. All of them, they have at least released 5 to 6 version of product in the market. One interesting material that I would like to share to all of you today is about the carbon. Very well that graphite, graphene, and carbon nanotubes, Buckminster fullerene, there are allotropes of carbon. So when you try to do a geometrical orientation, we have a zero-dimensional fullerenes, one-dimensional nanotube, three-dimensional graphite. This material, they have a very good electrical property, mechanical property. For example, single wall carbon nanotubes and MWCNTs, they have interesting properties. And these nanostructures can be very useful for the applications. CNTs, they have a multiple applications in the electronics, in the sensors associated with the biological system. Dear students and the delegates, I request you to note down these three elements: aluminum, magnesium, and carbon. Like energy and healthcare, these two sectors going to be in the research and in the deployment for the upcoming 20 years. Materials, they are going to be very supportive: aluminum, magnesium, and carbon. And we really require a lot of research people and research support for this three domain. For example, aluminum, magnesium, and carbon. If you are interested in doing this research in these three materials, we are open for collaboration in India, and this would be very interesting to have a joint research collaborations in these three domain particularly. For example, this is one case, one research that I would like to present to our students, that how the nanotubes can be fabricated. And this is a nanotube based light emitting devices. We can able to coat a gold in the edges, and by varying the tube diameter, we can able to get the photons being ejected. So light emitting diodes, there is a recombination of holes and electrons, and these CNTs, they are being useful in the optoelectronics. Apart from that, we have other applications of nanomaterials in the defense, in the military purpose, fabricating machine tools with the civil engineering structures. An example for a super hydrophobic coating, fire retardant and self-cleaning applications. And in the case of aerospace, I don't want to go much detail into this application, but it's very, very huge. Apart from that, there are a lot of products are available in the market. For example, self-cleaning, anti-icing, fogging products, antimicrobial, UV protection products, corrosion resistant, and waterproofing. So there are a lot of products based on nanomaterials, or at least a nano process being involved in the market. In going forward, there is one interesting domain that is really coming up, that is really needed for the fabrication of these biomedical devices, such as lab on a chip. That is nothing but nanoelectronics, that I would like to present for few minutes. In the earlier times, there was a statement that 640 kilobyte of memory should be good for everybody. But now we have a mobile phones wherein we have a 4 GB or 6 GB RAM, and there is a world market for 5 computers, but now each and every home we have minimum of 4 computers. For studying purpose, we have a laptop, we have desktop, and even our mobile phones are computers. So there is this is a evolution that happens. For example, in the year 1970s, Intel 4004 chipset were developed, where it had only few transistors and electronic devices, whereas now we have close to 22 billion devices being integrated. And this is the one development due to the material science, and in particular with the physics and the material science, which contributed for the development of the electronic devices. So this is the very first transistor. AT and T Bell Laboratories at the United States of America, they have developed this germanium based transistor. But I'm going to introduce a transistor called single electron transistor. Don't get confused that this single electron transistor works with only one electron. No, it is not so. This single electron transistor having the power to manipulate each and every electron. And this single electron transistor is with the help of quantum dots, or with the help of carbon nanotubes, that I can able to see here very well. At the same time, we have logic gates for a lot of applications, because when we have diodes, transistors, we can construct logic. So these kind of semiconductor logic gates can be changed with the help of chemical molecules, organic chemical molecules, wherein the chemical computers comes into the market, and we can able to exactly get the truth table from the chemical computers that we are developing. Not only this kind of devices, nanoelectronics, they play a huge role in getting the display. For example, this is a comparison of light emitting diodes with the various scales, length scale that I have given. But the LED that I am going to present is something new. So this is a conventional LED, but the LEDs which I am going to share is based on the nanowires. So this kind of LEDs, for to connect these LEDs, we need to do the soldering and so on. Whereas if we need to fabricate nanowire based LED, these LEDs, they are a crossbar architecture array, where these devices, they have been connected automatically. There is no need for the external connection. So there are 4 LEDs this side, 4 LEDs this side. So in a small region, we have a multiple LEDs, so the efficiency is very, very high. Not only this kind of nanowire based LED, we have a organic light emitting diode, or these are called as flexible electronics, or plastic electronics, or they are cheap electronics, or polymer electronics. they can be made very flexible because this is polymer which can be sandwiched between electrodes, and this can able to give a light emitting property. Not only that, we can able to have diodes and transistors constructed with the nanowires which find a interesting applications. When we have diodes, transistors and logic, we can go for a memory. So when we go for a memory, we will have a lot of applications. For example, with the storage that I would like to share, so we have a nanowire based diode, nanowire based transistors, we have a logic, and finally we can go up with the memory. One interesting memory storing devices that I would like to share is a MEMS, that is a micro electromechanical system, or NEMS, nano electromechanical system storage. So we have a media and read write tips, and we have these actuators that can able to make a movement of this system to be here and there. Apart from that, this be used in autonomous where a low power source is highly sufficient to make these devices to work. And I'm in the last part of the presentation that usually with the industries, the food industry that it is working, usually they employ manpower to do a check. This is a company which roast coffee and brew coffee. They produce coffee. So how this company test is that they employ manpower to check the flavor, to smell, to eat, and so on. So instead of that, if we could have a electronic tongue or a e-tongue, that is nothing but a nanomaterial based array. You can see very well here when we have a e-tongue, so it can able to do a pattern recognition of that. So not only with the biological application of electronic tongue, it is also possible for a artificial nose. So usually we get the smells of a flower or the fruit. In some cases, if we get the smell of a toxic gases, probably human beings, it's dangerous. So if we could able to develop a electronic nose, for example, to identify the gases from the seeds or to identify the gases from the environment, it would be very, very helpful. And research is going on in fabricating this electronic nose as well as electronic tongue. So these are the two different approaches to fabricate nanomaterials. Top down, a bottom up approach. Probably I will not go into this much detail, but I've given this to for your understanding. When we do cooking in our home, there are few interesting properties: the salt, spice, and tanginess, consistency. So these are the technical properties that we evaluate with our food. Whereas if we do the characterization or if we do the test with the nanomaterials, we have a lot of other properties like structure, morphology, topography, optical, electrical characterization that we need to do. Probably in 2 minutes, I will finish my presentation. I'm in the end of my presentation. So how we can able to view these nanomaterials? So usually with the millimeter scale, we use the human eye, whereas for the other system, we can use microscopes that contains electrons. We can use them, and we really need to understand this. We need to use these tools to see objects that are at the very, very lower scale. So as I had started, the first take home message that the transdisciplinary thing that nanotechnology is everywhere. It finds a lot of applications, materials, data storage, biological application, cosmetic, medicine, lab on a chip, and for the energy and healthcare. So with this, I end. Probably if I have time, the coordinators can inform a few interesting clips to show at least any one of these interesting creatures. It's your suggestion if I have time.
Okay, thank you very much for the presentation, Dr. Murthy, especially about the nanoscience technology. And now we know that nanotechnology is everywhere. Okay, well, ladies and gentlemen, maybe any question from the participants? You can write your question in the chat box, or you can use the raise hand features in the Zoom meetings. And then you can ask directly to Dr. Murthy. Okay, any question?
or if the participants would like to see any interesting clip with these 4 insects, a cockroach, a lizard, a chameleon, and a spider, you can say in the chat box or to the coordinator. I will play a video on these creatures to say how nanomaterials are involved. Participants also can share your opinion to see a clip on this, any one clip. I think we don't have time. At least any one of the clip from these creatures. Moderator can help whether we can go for only one video with this.
Okay, maybe we only have about 10 minutes, Dr. Murthy.
Okay.
Maybe you can play the video while
Yeah.
for
So
the.
which one shall we give it to the audience? A chameleon?
It's up to you.
Ah, okay. So
Which
probably
one do you think the most interesting one?
the most interesting one. I will try to do as soon as possible with this 10 minutes. I'm seeing the time. I will open it very fast.
Okay, thank you.
So that the people, yes. So I'm sharing a video on chameleon. So this is the chameleon that is with the in the bark. So just see that it is entering with the color. And now you see that it tries to change the color. So this is the nanocrystal on the skin of the chameleon. It changes to pink. This chameleon changed to almost 16 colors in 3 minutes. So it changes to green. And you can, this is green with another shade that you can see. And now this is another green, and see the transition with green to blue that the chameleon is making the change. And this is it. So you can find a lot of interesting clips that understands with the nanoscience and technology in the websites. So I'm open for the questions.
Okay, very interesting video about
Thank you.
the changing of color in chameleon. And maybe if there are participants here who feel interesting with this video or Dr. Murthy talk, you can ask or you can write it in the chat box. We will wait. Okay, any question? Okay, well, Dr. Murthy, I think there is no question from the participant because of the limitation of time also. We will go to the parallel session after this. And I would like to say thank you very much for Dr. Murthy for the presentation.
Thank you very much.
Hopefully all of us can get a new valuable knowledge, and for Dr. Murthy, maybe hopefully in the next time we can have a collaboration or joint research.
Sure. Thank you very much.
And maybe next time you can visit Indonesia. If
Yeah.
the COVID disease has over.
Sure.
Okay, let we give a round of applause for Dr. Murthy Pichumeni. Okay.
Thank you.
Doctor. Thank you, Dr. Murthy. Nice presentation.
Okay, Professor David.
Thank you, Professor Dafik. Your presentation also very interesting to the audience, getting the understanding of STEM. That is very, very important. That is very, very nice topic that you have presented. This is very interesting.
Thank you. Thank you.
Thank you, Professor Dafik.
Thank you, Doctor.
Okay, thank you very much. I will say it again for Dr. Murthy Pichumeni. This is the end of the plenary session in this first day of ICOPAMBS 2021. As a moderator, I would like to ask for your apologies if there are any mistake during this plenary session. And for the next, I will give it back to the Master of Ceremony. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much for all the speakers. Hopefully we get a new knowledge and new inspiration so that we can create some papers with a great quality. Ladies and gentlemen, the plenary session for today has been done. For participants, link certificate will be given at Sunday, so please stay tuned till the end of the ICOPAMBS 2021.
The next session is lunch break and parallel session. Parallel session will be held at 13:30 or 13:30. For all presenters, we really need you to change your Zoom screen name. For example, if your ID paper is 1, please change into paper ID 1 underscore your name. So the operator can move you to the right virtual room.
Pengumuman untuk peserta paralel akan dimulai pada pukul 13.30 waktu Indonesia Barat. Untuk seluruh presenter, dimohon untuk mengganti nama di Zoom sesuai dengan paper ID. Jika Anda memiliki paper ID 1, maka dapat dituliskan 1 underscore diikuti nama Anda, agar kami para panitia bisa memasukkan Anda ke dalam room masing-masing.
Berikut merupakan panduan dari pengubahan nama Zoom-nya. Jika paper ID Bapak Ibu adalah 1 atau 2, silakan diganti nomornya 1 underscore nama Bapak Ibu. Kemudian silakan menunggu, nanti kami akan membuat virtual room yang ada di Zoom, yaitu breakout Zoom, supaya nanti Bapak Ibu bisa masuk ke dalam virtual room yang tepat. Kemudian nanti kami akan memulainya jam setengah 2. Kemudian apabila Bapak Ibu sudah mengirimkan video melalui Google Form, dimohon Bapak Ibu tetap masuk di dalam breakout room untuk menjawab pertanyaan-pertanyaan dari peserta yang lain.
Thank you for your attention. We will held at 13:30 or 13:30. Thank you.
Baik, kita akan kembali lagi pada pukul 1:30 siang ini. Kemudian dimohon untuk dapat kembali join di plenary session di hari Minggu, karena pembagian sertifikat akan dilakukan apabila Bapak Ibu mengikuti kegiatan hari Sabtu dan Minggu ini secara terus-menerus. Terima kasih.
Recording stopped.